of infectious diseases. Translational research from bench to bedside to the field. The term epidemiology aims to study the prevalence, determinants, and prevention of a disease across the populations. Despite beneficial effects of traditional epidemiological studies on the prevention and control of many diseases, there still remain important questions about the biological mechanisms underlying various infectious diseases. In cases of infectious diseases, scientists have always been curious about the answers to the following questions, like what is the cause and pathogenesis of an infectious disease? Why do people respond differently to a specific pathogen, therapy, or vaccine? For example, why did some of the people present with severity in COVID while others stayed asymptomatic? And also how to effectively control the transmission of an infectious disease, and many more such questions. Gaining an in-depth insight into these questions is necessary for developing more effective prevention and control strategies. However, using traditional epidemiological studies alone, it is difficult to gain deeper understanding of the above questions, and hence the present workshop. Molecular epidemiology is a branch of epidemiology developed by merging molecular biology into epidemiological studies. Since its integration into the scientific world, the molecular epidemiology has provided valuable information about infectious diseases, prevention and therapy, and also the techniques have been widely used to cover knowledge gaps in research areas. With this brief information, let's seek the divine blessings of Lord Ganesha, who is the remover of all obstacles, through an invocation song. I now request Ms. Gayatri and Ms. Yashaswini to render the invocation song.
ಎಲ್ಲರೂ ಸೇರುವ ಶುಭ ದಿನವೂ ಬೇಗನೆ ಬಾರೋ ವಿನಾಯಕ, ಬೇಗನೆ ಬಾರೋ ವಿನಾಯಕ. ಎಲ್ಲರೂ ಸೇರುವ ಶುಭ ದಿನವೂ ಬೇಗನೆ ಬಾರೋ ವಿನಾಯಕ, ಬೇಗನೆ ಬಾರೋ ವಿನಾಯಕ. ಪರಮೇಶ್ವರ ಪ್ರಿಯ ಪುತ್ರನಾಗಿಹ ವಿಘ್ನರಾಜನೇ ಪಾಡುವೆವು, ಪರಮೇಶ್ವರ ಪ್ರಿಯ ಪುತ್ರನಾಗಿಹ ವಿಘ್ನರಾಜನೇ ಪಾಡುವೆವು. ಶ್ರೀಲಂಬೋದರ ಸೇತಿ ವಿನಾಯಕ, ಶ್ರೀಲಂಬೋದರ ಸೇತಿ ವಿನಾಯಕ, ಜ್ಞಾನವ ನೀಡೋ ಗಜಾನನ. ಎಲ್ಲರೂ ಸೇರುವ ಶುಭ ದಿನವೂ ಬೇಗನೆ ಬಾರೋ ವಿನಾಯಕ, ಬೇಗನೆ ಬಾರೋ ವಿನಾಯಕ. ಕುಮಾರಸ್ವಾಮಿಯ ಸೋದರನಾಗಿಹ ಶರಣು ಶರಣು ಶ್ರೀ ವಿಘ್ನೇಶ, ಕುಮಾರಸ್ವಾಮಿಯ ಸೋದರನಾಗಿಹ ಶರಣು ಶರಣು ಶ್ರೀ ವಿಘ್ನೇಶ. ಪಾಪವಿದೂರ ಪರಮದಯಾಕರ, ಪಾಪವಿದೂರ ಪರಮದಯಾಕರ, ಮುನಿಜನ ವಂದಿಪೆ ನೀ ತೋರೋ. ಎಲ್ಲರೂ ಸೇರುವ ಶುಭ ದಿನವೂ ಬೇಗನೆ ಬಾರೋ ವಿನಾಯಕ, ಬೇಗನೆ ಬಾರೋ ವಿನಾಯಕ.
Thank you, Ms. Gayatri and Ms. Yashaswini. I now request Dr. Asha Srinivasan, Assistant Professor, Division of Nanoscience and Technology, School of Life Science, JSS Academy of Higher Education and Research, to kindly welcome the gathering.
Good morning, everybody. Good morning, everybody. Seeking the blessings of His Holiness Jagadguru Sri Shivaratri Deshikendra Mahaswamiji, I, Asha Srinivasan, from School of Life Sciences, invite this august gathering for the 7-day INDO-US workshop on molecular epidemiology of infectious diseases, translation research from bench to bedside to the field, at JSS Academy of Higher Education Research, Mysuru, in association with PHRI, which is Public Health Research Institute of India, Mysore, and synergistic training program utilizing the scientific and technological infrastructure, in short, STUTI, under the DST DST program, Government of India. We have our participating institutes, University of California, Berkeley, University of Arizona, Tucson, and University of Toledo, Ohio, and NIH Fogarty International Center. I, along with the organizing committee, organizing chairman, Dr. M.N. Suma, sorry ma'am, and organizing secretary, Dr. Subbarao, welcome our Honorable Pro-Chancellor, Dr. B. Suresh, Honorable Vice Chancellor, Dr. Surendra Singh, our inspirational leaders. They have not been able to attend today, but they have conveyed their best wishes for the success of this week-long workshop. As already said, the main purpose of this workshop is to align basic research to translational and, of course, give a hands-on workshop to the delegates, to the participants. I welcome our registrar, Dr. B. Manjunatha, who has been a pillar of support for all academic and research endeavors. I warmly welcome Dr. Kushalappa, Director Academics, Dr. Prashant Vishwanath, Director Research and coordinating DST Stuti program. I welcome Dr. Basavanagodappa, Principal, JSS Medical College, who has always extended his support in all science and technology activities. I welcome our US delegates. First, Dr. Purnima Madhivanan, Associate Professor in Health Promotion at Mel Ednet Zuckerman College of Public Health at University of Arizona. Dr. Purnima is also the founding director of Health Research Institute of India, PHRI, Mysore. Next, I welcome Dr. Kerry Cooper, Assistant Professor, Food and Safety and Epidemiology, the School of Animal and Comparative Biomedical Sciences, College of Agriculture and Life Sciences, University of Arizona. Dr. Kerry is also the co-director of the University of Arizona's One Health Consortium. Welcome. I welcome Dr. Carl Krupp, Assistant Professor in Public Health Practice and Translational Research at the Mel and Ed Zuckerman College of Public Health at University of Arizona, Phoenix. Welcome, sir. I welcome Dr. Matam Vijay Kumar, Professor at Physiology and Pharmacology, University of Toledo. One of the pioneers, Dr. Vijay Kumar is one of the pioneers in discovering the links between gut microbiota and metabolic syndrome. Welcome, sir. I welcome our today's inauguration personnel, Dr. Anjali Arun, Managing Trustee, PHRI Mysuru. Ma'am, thank you for agreeing to inaugurate the function. I welcome all the chairpersons of this INDO-US workshop, resource persons for all the sessions. Welcome and also welcome the organizing team. I welcome Dr. Sri Satish Chandra, Administrative Officer. I welcome all the delegates. This INDO-US workshop is special because for you. Okay, it's for you it is made. So I welcome all the delegates from various parts of the India and a very, very warm welcome to all of them, teaching and non-teaching staff and all the helping members. I wish you all the very best and a productive and pleasant stay in Mysore. Once again, on behalf of all organizing committee, I welcome everyone for the INDO-US workshop. Thank you.
Thank you, ma'am. It is not what is poured into a student that counts, but what is planted. With a set phrase, I request all the dignitaries to kindly inaugurate this week-long workshop by watering the plant. Thank you all. I request Dr. Anjali Arun, Managing Trustee, Public Health Research Institute of India, Mysore, to kindly deliver the opening remarks.
Good morning and namaste to everybody. I welcome all of you for this workshop and the dignitaries on the dais, those who are present and those who have blessed us in absentia. All of them I welcome. And this workshop is a great thing. Like a common person, I just decided to, look at the Google of molecular biology and infectious disease. The term molecular epidemiology, was first used sometime in 1973. So in about 49 years, huge progress has been made. And when I put molecular epidemiology and COVID, it is some 6 crore articles come up, 6 crore and several lakhs. And I say molecular epidemiology plus COVID, 4 crore plus articles come up. That means that this is the state of the art and very, very cutting edge technology that you are going to learn over here and definitely make contributions to public health. Talking about public health, I must not miss this opportunity to thank ICMR and our government for the timely and practicable guidance and policies that they have made. Compared to the so-called developed countries, our country has really given very, very wise advice to the people which was followable as compared with the other countries who have leveled draconian measures or been too lax. I think we have done pretty well in a disease where the whole world has actually made mistakes. It was expected that India would make a lot of mistakes, but at this point in time, I would like to say that we have done well as far as COVID is concerned. And so for future, I think we will be able to offer as a scientific committee good inputs to the government and to the policymakers and to the public health personnel with the help of all the work that you all will go across the country and probably internationally. Do it, and most important, collaborate with each other to share the information and further the interest of scientific research. So with these few words, with the divine blessings of Shri Desikendra Swamiji, I declare that this workshop is open. Thank you. I would like to thank the organizers for giving me this opportunity to be here in this August gathering. I do not consider myself particularly qualified to actually talk in a gathering like this, but I am an important part of the diagnostic industry, and I have been so for the past 30 to 35 years. And the collaboration between the diagnostic and the research is very much what is required today. Research should not be isolated. Diagnostics and clinical areas should not, shun research. From the bench to bedside and to the field should all merge actually, and it should take place simultaneously. Thank you.
Thank you, ma'am. I now request Dr. Prashant Vishwanath, Director Research, JSS Academy of Higher Education and Research, to kindly enlighten us about the workshop.
Just before, sir, just 2 minutes. Sorry, I forgot. We welcome Dr. Lee Rayleigh from University of California, Berkeley. Welcome, sir.
A very good morning to all. Greetings from the JSS Academy of Higher Education and Research, and welcome all of you to this INDO-US workshop being organized by the Department of the initiative being taken by the Department of Biochemistry, JSS Medical College. This workshop is a 7-day workshop on mainly molecular epidemiology of infectious disease, translational research from bench to bedside. And in this workshop, we have got participants from Pan-India. The time was very short for us to invite participants from outside the institution as well as outside the state. This workshop was being organized by Dr. Subrao and his entire team, and all of you, many of the young team who is here, they were part of the organizing team. We were fortunate to get major funding from the Department of Science and Technology in the form of a Stuti grant that is mainly for synergistic training and utilizing the technological infrastructure which is available at the DST recognized FIST centers across India. We were one of the 13 institutions to get this important project. And through this, we have to conduct training and awareness programs for the benefit of the society and the young researchers. So we successfully conducted the awareness program during the 22nd to 28th of February. It was a National Science Week, and we had a very great response for that awareness week, Science Awareness Week. And we were immediately asked to conduct a training workshop, and then we had to do a little bit of modification for this particular workshop and see that it aligns with the objectives of this Stuti. And but still, we are happy that we have got about 15 participants who are from different regions of the India. We have got participants from Delhi, South, Chennai, and many other regions. So it's heartening to invite you here and see that you benefit from this workshop. So there are a lot of, this workshop is actually a blend. It's a blend of knowledge as well as skill. Knowledge in terms of, we've got very highly renowned United States, as well as we do have a workshop mainly on the molecular methods, as well as some advanced techniques like confocal microscopy as well, right? So it's a perfect blend, I feel, and I'm confident that you all will find this workshop useful, and you would look forward for further such kind of training programs being offered from our institution. Thank you very much. Once again, I welcome you all. Thank you very much.
Thank you, sir. I now request the dignitaries to please release the conference chronicle and workshop manual.
So what you will have? Oats only. Afternoon?
Rice, sir? Yeah. Oh, Monday.
I now kindly request Dr. M.N. Suma, Vice Principal, JSS Medical College, to introduce the resource persons to the gathering.
Thank you, Dr. Kusuma. Very good morning to all. On behalf of the organizing committee, it gives me an immense pleasure in introducing international delegates of this workshop, which is mainly focused on molecular epidemiology of infectious diseases, translational research from bench to bedside to the field. Firstly, we have with us Professor Lee Relly, who has joined online, and he is head of Division of Infectious Disease and Vaccinology, School of Public Health, University of California, Berkeley. Sir has pursued BA from Stanford University, MD from University of California, and he has done residency program in internal medicine. He has served as Epidemic Intelligence Service Officer at the Center of Disease Control and Prevention. He is a project manager for the World Health Organization in New Delhi, India. He is member of Board of Scientific Counselors for the Center of Disease Control, and he has published more than 280 peer-reviewed papers to date and 2 books. It's mainly on molecular epidemiology of infectious diseases, principles and practice, and slum health. And sir will be explaining about molecular epidemiological aspects in this workshop. We are happy to have you here online, sir. We will welcome Dr. Lee Relly to this workshop. And we have Professor Purnima Madhivan, who is adjunct faculty of JSS Academy of Higher Education and Research. She is Associate Professor in Health Promotion Sciences at Mel and Ellen Zuckerman College of Public Health, University of Arizona. She is physician trained in India with addition of MPH and PhD in epidemiology. She is Director of Global Health Training Programs at University of Arizona in collaboration with Stanford, Yale, and University of California. She directs Fogarty Fulbright Fellowship Program for University of Arizona, and she is founding director of Public Health Research Institute, Mysuru. Her work is mainly focused on disadvantaged populations, illustrating the dynamics of poverty, gender, and environmental determinants of health, in particularly impact on women and children in rural communities. And she has received prestigious leadership award from the Elizabeth Glaser Pediatric AIDS Foundation for her work on HIV prevention. And she'll be speaking on basic epidemiological concepts in this workshop. And on behalf of organizing committee, I welcome you, and I really thank you for accepting this invitation. The next speaker we have with us here is Dr. Carl Krupp. He is Assistant Professor in Division of Public Health Practice and Translational Research in Mel and Ellen Zuckerman College of Public Health, University of Arizona. He is involved in public health services in disadvantaged communities in US and India. He has been working in India on social determinants of health and non-communicable diseases and raised several public health foundations in India, US, and Europe. He has mainly worked on HIV prevention, maternal health, primary and secondary prevention of cervical cancer, vaccine hesitancy, and cardiovascular diseases. Dr. Krupp has written more than 50 successful grants to US National Institutes of Health and other multiple foundations in Europe and India. He has raised $5 million over last decades and has 106 publications to his credit. And is here with us to share his expertise on grant writing and scientific manuscript writing. On behalf of the organizing committee, I welcome Dr. Krupp for this workshop. The next speaker we have here is Kerry Cooper, Dr. Kerry Cooper. He is Assistant Professor of Food Safety and Epidemiology at School of Animal and Comparative Biomedical Sciences, University of Arizona. He has done his BS in Biology and PhD in Pathobiology. He has served as Environmental Health Specialist. He is co-director of University of Arizona's One Health Consortium. His research lab focuses on epidemiology, genomics, and pathogenesis of foodborne pathogens. He is expert in next-generation sequencing to develop low-cost, rapid, portable, and accurate methods for detection of prevention of foodborne pathogens in global food supply. And he'll be sharing his experiences on molecular epidemiology, next-generation sequencing, and bioinformatics in this workshop. On behalf of the organizing committee, I welcome you, Dr. Krupp. Next speaker we have with us is Mysorean, Dr. Nathan Vijay Kumar. He is alumni from CFTRA. He is Professor of Physiology and Pharmacology, Medical Microbiology and Immunology, University of Toledo, and he is Director of University of Toledo Microbiome Consortium. His areas of research is mainly on host-gut microbial interactions, and he is member of prestigious organizations like American Association of Immunology, American Society of Nutrition, and many more. His discovery of the link between gut microbiota and metabolic syndrome but has earned many accolades in the global scientific community. He'll be sharing his experience on host-innate immunity and gut microbiota in health and diseases. And on behalf of this committee, I welcome Dr. Nathan Vijay Kumar. So friends, we have scientifically rich team from US, and we'll have a fruitful research collaborations once this workshop is over. And I request all participants to make best use of this opportunity and progress well in research field. Thank you all. Thank you all.
Thank you, ma'am. I now request Dr. Tonima Madhivanan, Associate Professor, University of Arizona and Founding Director of Public Health Research Institute of India, to give a few comments regarding the program.
Good morning, everybody. Namaste. Welcome, everyone. I want to first start off by thanking the Swamiji and the organizers for bringing us all together here as a group to learn what is going to be the next generation of science and research that's going to happen in this world. If I may ask, how many of you are below 35 years of age?
Many.
Well, you are the future leaders, and we are here today to support you in actually doing what we hope we could achieve in our lifetime. We're all living in an era of globalization, and whether we like it or not, it's going to change the way we practice science and epidemiology. The kind of questions we ask and the methods we use to answer them. Increasingly, the public health problems are being shifted from rich countries to the poorer countries, and from rich populations to poorer populations within the Western countries. There is increasing interest and concern about situation in the non-Western populations on part of the Western scientists and epidemiologists with regard to collaborative research, skills transfer, and the idea of volunteerism to enable benefits for the Western world, and the approaches to epidemiology to be shared by the non-Western world. The most existing collaborations that we have usually benefit the Western world. And we are hoping that these kind of partnerships and these kinds of workshops will change that. We want to move that balance from having everything be of benefit to the West to actually happen here, because we are the next generation of leaders. And that's why I asked how many of you are below 35 years of age. Even when research in non-Western populations is conducted as a genuine collaboration, it's too often an export failure from the West. We take a Western concept and we think that it will work in our cultures and in our communities. And most times that fails. And we have already seen that happening with the aid programs, all the grants that come with the idea of adapting a Western model to our communities. They never work. So what we want to do is our scientists, our leaders here have a lot to contribute to the world. And the future is with us here. And we need to embrace that, get confident about it, and make sure that we all have a seat at that table when discussions and decisions are being made. And that would also include the recognition of the importance of context and the importance of diversity and local knowledge and the problem-based approach to addressing major public health problems using the most appropriate technology. The development of a truly global partnership in science and epidemiology can not only better address the public health problems in the non-Western populations, but it can also shed light on the current limitations of epidemiology in addressing these problems, just as Dr. Anjali said. We have something here, and let us share it with the world. And these kind of workshops are not for us to come to tell you what to do, but for us to learn from you and for you to learn from us. We want to have the true partnership. And with JSS, I was thinking, how long have I known my colleagues in JSS? There was a saying Henry Ford said, coming together is a beginning. Keeping together is progress. Working together is success. We started collaborating with JSS sometime in 2011. And in partnership with Dr. Lee Riley, who's here, who should actually be speaking, and I am only representing Dr. Riley here today, and hopefully next time it will be Dr. Riley who's going to be addressing you. We started a very, very, very small project here with collaboration with Dr. Anjali at Public Health Research Institute of India. And fast forward today, here I have colleagues, Dr. Subbarao, Dr. Mahesh. If I keep saying I'm going to take up all the time, but I've had such amazing collaborations here at JSS, and we only want to make it stronger. We want to make it better. And we hope that this is just the beginning of a long, long, long successful road for all of us to learn from each other. Thank you.
Thank you, ma'am. To conclude the inaugural program, I request Dr. M.B.S.S.T. Subbarao, Professor and Organizing Secretary, JSS Medical College, to deliver the vote of thanks.
With the blessings of His Holiness, on behalf of the organizing committee, I take this opportunity to propose the vote of thanks. First, I would like to thank the resource persons who have traveled all the way from United States to come and conduct the workshop in JSS. Dr. Punima Madhivanan, Dr. Karl Krab, and Dr. Kerry Cooper, and my friend, Dr. Gautam Vijay Kumar. I thank you all. Even though he is not present in person, Dr. Lee Riley has helped us a lot, beginning from day one, in the preparation of the schedule and all the way to the preparation of the working modules for the modular exercises. He has helped us a lot. He has guided us. In addition, he has also supported this workshop by providing the funds from GID grant. So I thank Professor Lee Riley for all the support he has extended. Thank you, Professor Lee Riley. I also thank all the chairpersons who have agreed kindly to chair the sessions of this workshop. I thank all the leaders of JSS AHER, in particular our Pro-Chancellor, Professor B. Suresh, Vice Chancellor, Dr. Surinder Singh, Registrar, Dr. B. Manjunatha, Director Academics, Dr. P. A. Kushalappa, Principal of our college, Dr. H. Baswanagaudappa, our Vice Principal and Head of the Department, Dr. M. N. Suma, our Director of Research, Dr. Prashant. Dr. Prashant also has extended the financial support from the Stuti grant. I thank.
Thank you.
I thank the people who have directly or indirectly participated and supported this workshop. And our sincere thanks also to the Chief Information Officer, Dr. Ravindra, and his team for all the work that they have carried out and helped us to live telecast this program. I also thank our administrative officer, Sri Satish Chandra, and the project coordinator, Shilpa, from the JSS Medical College. office. I also thank the support staff from the JSS Medical College and our Department of Biochemistry. And my sincere thanks are also due to the funding agencies, that is DRDO, and some equipment companies like AAN Scientific and the SciEx, who have supported this workshop by providing some funds. I extend my sincere thanks to my colleagues, department members, teaching and non-teaching staff, and all the research scholars of the CMR lab and students. Finally, I thank all the registered participants who have come from different parts of the country to attend this workshop. Hope you enjoy this 1 week long stay in the Mysore and gain some knowledge in this workshop. Thank you all.
Thank you, sir. We will now begin with our first session on principles of molecular epidemiology. I invite Dr. M.N. Sumana, Professor, Department of Microbiology and Director, Laboratory Services, JSS Hospital, and Dr. N.B. Ramachandra, UGC DSR Fellow, Department of Genetics and Genomics, University of Mysore, to chair the session. I request the chairpersons to introduce the speaker, Dr. Lee Riley, to the audience.
Riley has been introduced already, no?
Good morning, everyone. Dr. Sumana, Professor of Microbiology and Director of Laboratory Services. It's my pleasure to introduce Dr. Lee Riley, Professor and Head of the Division of Infectious Disease and Vaccinology at the School of Public Health, University of California at Berkeley. He served as an Epidemic Intelligence Service Officer at the Centers for Disease Control and Prevention, Infectious Disease Postdoctoral Fellow at Stanford University School of Medicine, Project Manager for the WHO organization in New Delhi, India, and a faculty member in the Department of Medicine at Cornell Medical School in New York City. He joined the faculty of the School of Public Health at the University of California, Berkeley in 1996. He was a member of the Board of Scientific Counselors for the Centers for Disease Control from 2014 to 22, and is a fellow in the American Academy of Microbiology. His research focuses on the molecular epidemiology of infectious disease as well as diseases of the urban slum communities. He collaborates with researchers in Brazil, Colombia, India, Japan, China, and Bangladesh. As a person of infectious disease specialty, it's my pleasure to present before you Dr. Lee Riley. Thank you. Thank you, one and all.
Thank you. Thank you, everyone. Can you hear me? Can everyone hear me?
Yes, sir. Yeah, we can hear you.
Okay, great. So it's a real pleasure to be able to do this. Unfortunately, I cannot do this in person. I had a very special, as Dr. Madhivanan knows, I have a very special reason why I could not attend the session in person. I had a new member of a family join us. His name is Max. He was born 3 months ago. They put to help my wife out. Hopefully, there'll be another occasion, maybe next year, to do this again, where I could attend the workshop in person. As Dr. Madhivanan just mentioned, I have been to Mysore several times. In fact, it was Dr. Madhivanan who introduced me to her PHRI organization, as well as many of the faculty at JSS University, with whom we've been able to put together this workshop. And so my relationship with JSS University and Mysore goes back many years. So even though this is called the Indo-US Workshop on Molecular Epidemiology of Infectious Diseases, we probably should title it the first Indo-US workshop on molecular epidemiology of infectious diseases, because I anticipate that we'll be doing several more of this workshop in years to come. As just mentioned, I work in Brazil. I've been working in Brazil for more than 25 years. And we also do a one-week workshop on epidemiology there. And we first started this workshop in Brazil. I meant to do it just for 5 years, and then the Brazilians were supposed to take over. Didn't happen. This workshop has been continuing every year for 20 years straight. It was only interrupted the last 2 years by the pandemic. So hopefully we can do the same for this Indo-US workshop, particularly to be based in Mysore. So with this background, let me begin. So I think several people mentioned the importance of the new technology that's applied to do epidemiology, but I'm going to emphasize not so much the technology, but the basic principles of molecular epidemiology. I will talk briefly about techniques, but what's really important is really understanding the basic principles of molecular epidemiology. There's a lot of confusion out there, even in the literature, of what constitutes molecular epidemiology. I'm going to start out by presenting an overview of this concept and some of the definitions that I used. And I guess my slide is shared. Everybody can see the slides? Okay. So, to start with, of course, I mean, I can't give any talk without talking about the COVID situation. And very early on when the pandemic began, people asked these types of questions. Where did this virus come from? In the case of US, we asked the question, when did it enter the United States, Europe, Brazil? And we can ask the same question, when did it enter India? Will it go away completely like SARS-CoV-1? First SARS-CoV pandemic. This is something that happened years ago, and after about 6 months, it just completely disappeared. So this is one of the questions that we asked early on, and today we say that no, it has not gone away. It's continuing. Will it become a seasonal disease like the influenza virus? Well, not yet. I think it's still too early to tell. Why is it so transmissible? Can a vaccine be made against this virus? So as I mentioned, this question was asked at the beginning of the pandemic. We now know that there are many vaccines that have been developed against this virus. And so these are kind of questions that come up that can be addressed by molecular epidemiology methods. And in fact, all of these questions are answered and addressed by new molecular epidemiology methods. So let me just go into some of the basic concepts. So some of the answers required in molecular epidemiology approaches. So what is molecular epidemiology? The first paper that I saw in the literature that used the word molecular epidemiology in the title was this. This appeared in 1980 by Huang et al. And it said, the title of the paper said, molecular epidemiology of cytomegalovirus infection in women and infants. And this was a New England Journal paper. This was the abstract. It said, we studied CMV isolated mothers in their children to determine whether recurrent infections, transmission to fetus in immune women are due to reinfection or reactivation of endogenous virus. Endogenous CMV appeared to be the most frequent source of recurrent infection and intrauterine transmission in immune women. Infection also occurs, but less commonly. So this is actually a very good example of what a molecular epidemiology study is. It addresses this basic question of the reinfection that occurs in CMV infections. And that asks the question, is the reinfection is the transmission due to reinfection with a new virus strain or reactivation of the endogenous virus? And this is something that comes up all the time in many other infectious diseases, such as tuberculosis and HIV. This is it's a fundamental question that comes up, and this can only be addressed by molecular epidemiological methods. So, if you were to be more exact about the definition of molecular epidemiology, so the definition of epidemiology that everybody's probably heard of is the study of the distribution and determinants of distribution of disease in human and also non-human populations. In the past, I only included the word human, but when I give this talk in many other places, there are inevitably veterinarians in the audience, and they complain that I didn't include non-human animals. So I put this in. Yes, epidemiology does apply to non-human populations as well. So the molecular epidemiology is really an extension of this definition. So it's the study of the distribution and determinants of distribution of infectious diseases using molecular techniques. So that's a simple, straightforward definition of molecular epidemiology. But there's also another important definition that we can use, and this is the study of the genetics of pathogens that determine disease transmission. This is what we do in our lab. We're interested in the basic genetics of how organisms, how microbes transmit themselves to cause disease in humans. And so the important word in the definition is the determinants of distribution. This is what we study. It's important to, of course, study the distribution, but in molecular epidemiology, we really focus on the word determinants. And then we also came up with a new term that we call next generation molecular epidemiology. So this is the study of the distribution and determinants of distribution of infectious diseases using next generation sequencing. So as was mentioned several times, the technology, the molecular microbiology technology has really evolved in the last 10, 15 years. And it's very important for epidemiologists to keep track of these new developments in the technology used in molecular microbiology, and try to understand how technology can be used to address epidemiological questions that cannot be answered by traditional laboratory methods or traditional epidemiology. And then, so the other component of the next generation molecular epidemiology is something that I think Dr. Vijay Kumar is doing. This is the study of complex microbial niches that determine infectious and non-infectious diseases. So unfortunately, this 1 week will not be enough to really go into these some of the examples that illustrate these points. Here at Berkeley, I teach this course. It's a one-semester course. So we're trying to condense this one semester course into just less than 7 days. And so cannot do justice to all of the issues that we need to discuss. But at some point, we could do this. But if you want to learn more about this, you can come to Berkeley and take my class. So let me just ask you this. I pulled out several papers that have been published with the words molecular epidemiology in its title. So here's one. It says molecular epidemiology of the skill streptococcal invasive locus in group A streptococci causing invasive infection in French children. This is the abstract. 31 different EMM toxin genotypes among 74 group A streptococcal isolates causing invasive infections in French children. Predominant EMM types were EMM1, EMM3, EMM4, EMM6, EMM89. 16% of isolates harbored the streptococcal, half of them belonging to EMM4. Is this molecular epidemiology? What do you think? I see the group here from my computer screen. So how many of you think this is a molecular epidemiology investigation? Please raise your hand. Okay, maybe it's the way I'm asking the question, so nobody's raising hands. Okay, so if you're not saying that this is molecular epidemiology, then what is this? What is this? And don't be shy. You can, I'm going to ask a lot of questions during this week, so you can answer the questions. So what kind of study is this if it is not a molecular epidemiology study? Okay, so let me answer this for you. So this is, I would not call this a molecular epidemiology study, because if you look closely at the abstract, it only talks about the bacteria, the streptococcal organism. It doesn't say anything about people. It doesn't talk anything about the individuals who get infected with this organism. Epidemiology is about people and non-human animals. It's not about organisms. So all they did in this study was genotype this streptococcal organism, but we don't know anything about the children, these French children. We don't know about anything about their age. We don't know about their gender. We don't know about where they come from. We don't know where they live. We don't know anything about these French children. So this is not an epidemiology paper. This is not molecular epidemiology. This is what I call a taxonomy study of streptococcal organism and its distribution. Okay, what about this? This is another study that appeared in Japan. It says molecular epidemiology of Mycobacterium tuberculosis in an urban area in Japan, 2002 to 2006. So the setting was Shinjuku City in Tokyo. Objective was to evaluate the status of transmission of Mycobacterium tuberculosis in Shinjuku City, allocate resources efficiently and effectively for successful TB control program. The study design is observational descriptive study combining genotype data of M. tuberculosis with TB patient. These are the results. The genotype clustering rate was significantly higher in males aged less than 40 years and homeless, and was lower for the foreign-born. Among 45 genotype clusters containing 152 TB patients, 26 clusters containing 102 patients were composed of a mix of homeless and non-homeless patients. The conclusion, the study revealed that M. tuberculosis transmission occurred more frequently among the homeless than in non-homeless persons. However, transmission by casual contact between the homeless and the general population was also shown to occur. Is this molecular epidemiology? Yes, definitely. We know a lot about the people in Shinjuku City, people who get TB. And so what they did here, which is something that I will go into later, is they were genotyping the Mycobacterium tuberculosis organism. And when they say cluster represents recent transmission of the organism. This is something that people in the field have accepted as a way to distinguish reactivation disease from recent transmission disease. So as you know, tuberculosis can occur in someone who's been infected for many, many years, reactivation TB, but it can also occur in someone who is recently infected. So you get infected and you rapidly progress to active disease. This is a very important question, because if most of the cases in a community are due to recent transmission, TB control program right now is not working. If most of the cases are due to reactivation disease, that means that the current TB control program may be working, but it's not working very well in the past. So this is a very important type of investigation to do, to genotype Mycobacterium tuberculosis to distinguish what proportion of new TB to reactivation versus recent transmission. And this is what they did. They found that most of the transmissions are occurring among the homeless population in the city. So this gave us a very important epidemiological understanding of TB in this part of Tokyo. Very good example of a true molecular epidemiology investigation. So molecular epidemiology has to be distinguished from other similar disciplines, such as phylogenetics. Phylogenetics is a study of lines of descent or evolutionary development of an organism. That's not the same thing as molecular epidemiology. Taxonomy. Taxonomy is the science of classification of organisms into natural related groups based on a factor common to each. Taxonomy is not the same thing as molecular epidemiology. Molecular evolution is also not molecular epidemiology. Molecular evolution is phylogenetics based on analysis of nucleic acid sequences per evolutionary relationships of organisms. So these disciplines, phylogenetics, taxonomy, and molecular evolution, they study the relationships of organisms to each other. Whereas epidemiology, we study the relationship of organisms to each other and to their hosts within some environmental context. And of course, right now, this microbiome is also playing a major role in this relationship, these components involved in the study, that can be studied by molecular epidemiological methods. This is the difference between molecular epidemiology and these other disciplines called taxonomy, phylogenetics, and molecular evolution. Now, of course, in molecular epidemiology, we use the same tools that are used in taxonomy or phylogenetics and molecular evolution. The tools are similar or the same, but the disciplines are very different. So if you're only typing organisms and then telling a story about the relationships of organisms, that is not molecular epidemiology. So this is a very important point that I want to stress from the very beginning. So in epidemiology, we generate hypotheses and we can test the hypothesis empirically. Epidemiology also provides opportunity for intervention. It's never technique driven. It's not technique dependent. Whereas phylogenetics, taxonomy, molecular evolution, they tend to be a descriptive discipline. Information is always inferred. Information is inferred is because in order to come up with some sort of phylogenetic tree or taxonomic model or molecular evolution model, you need to have enough samples. You need to get enough samples to really be able to come up with some sort of evolutionary model. And so one becomes really dependent on the samples that are available. And in many situations, if the samples are not rare, you really cannot come up with any good model. And there's no such thing as intervention in these disciplines. That's not what they do. The descriptive discipline, there's no intervention involved. It's always technique dependent. So what are some of the laboratory methods used in molecular epidemiological investigations? So the laboratory data are used to stratify data. This is the core principle in molecular epidemiology. We generate data using laboratory techniques to stratify data. For instance, we have some sort of microbial population in some cells, such as the blood or cerebrospinal fluid. I put the word sterile in quotation marks because blood and sterile spinal fluid, they're really not truly sterile. You do PCR, kind of random PCR, you can actually amplify all kinds of viral sequences in these samples, in these human specimens. And so, but nevertheless, we're looking for pathogens and try to look for the pathogenic organism in these sterile niches. If the microbial population is in a non-sterile niche, such as the end of the mouth, we have to then separate the target organism from the other organisms. And the point is to eventually come up with some sort of pure culture organism, or pure isolation. And then you identify the organism to the species. So this is what's initially done in most clinical laboratories to make the diagnosis of infectious disease. But in molecular epidemiology, we actually go further than just speciating an organism. And we'll talk about this shortly, that we actually have to get to clone, identify some sort of clone. So we separate. So in the viral infections, it's a little more complicated. For viruses, they must be the target virus has to be separated from some biological samples using things like filtration, nuclease treatment, ultracentrifugation, isolation by cell culture, independent diagnostic tests, and then they're speciated, just like the bacteria, or further phenotypic subtyping tests, such as serotyping, and then genotyping. And with viruses, we have to also worry about quasi-species identification. So let me just give you I want to use an example of salmonella. The classification, the taxonomic classification of salmonella has evolved over time. So before 1960, there were more than 1,000 so-called species. And so these organisms, salmonella, were classified based on the O, H, and VI antigens. This is called the Kaufman-White scheme. But from the 1960s through the 80s, salmonella became reclassified and belonged to 3 species: Salmonella typhi, Cholera suis, and Enteritis. This was based on biochemical reactions. This is known as the Ewing's classification. Currently, Salmonella is divided into 2 species, basically Salmonella enterica and Salmonella bongori. And this is based on the ribosomal basic. So Salmonella enterica, further speciated, further subtype into 6 subspecies. And then also serotypes, where there are more than 2,500 serotypes. Salmonella bongori was formerly classified as subspecies 5. But then the next question is, what is species? So in 1980, the number of bacteria that were ranked at the level of species was 1,791. By 2012, that increased to 9,620. And by 2017, 10,000. So what does this mean? Does that mean that during this period, people identified all these new species of bacteria? No, that's not what happened. What happens was a change in the definition of species. So there's a nice paper written by Dr. Janda and Abbott, so based on 16S ribosomal RNA sequences. So what they are suggesting is that strains with less than 97% similarity score are considered to belong to new species. Similarity score of more than 97%, it's unclear, there's no general agreement. So using this sort of a consensus definition, as of September 2016, there are more than 3 million ribosomal RNA sequences cataloged. There are some bacteria that cannot be easily accurately classified by 16S ribosomal RNA sequence. And as you can see, they include many bacterial organisms that are important to human pathogens. Pseudomonas, Bacillus, Rotella, Bacteria, Campylobacter. I mean, these are all potentially important human pathogens. And these cannot be accurately classified by 16S ribosomal RNA sequencing. So this is kind of the recommended guidelines for 16S ribosomal RNA sequencing. Bacterial organisms with known problems with 16S ribosomal RNA sequencing, one can also sequence other genes, such as RPOB. Minimum of 525 base pairs should be sequenced in the 16S ribosomal RNA, but ideally more. And the criteria for species identification, minimum more than 99% similarity, ideal more than 99.5% similarity, and then they should be matched to known species that have been tested by DNA hybridization. For matches of distance score of less than 0.05%, the next closest species, other criteria such as phenotype should be considered. Now, this is sort of the way was recommended back in 2007. Because we have this new, not new anymore, but we have this technology of whole genome sequencing. This is completely changing the way we think about bacterial species. And this has introduced this new term called pangenomes. How many of you have heard of pangenomes? Raise your hands. No? This is something new. So pangenome, instead of all genes within a species. So for instance, if you have an E. coli, if you sequence one isolate of E. coli, you identify about close to 4,000 distinct. But if you sequence the whole genome of a second strain of E. coli, you identify new genes that were not present in the first strain of E. coli. You keep doing this, so every time you sequence another strain of an E. coli, you identify new genes that were not present in the earlier strains. And so pangenome can be further divided into these following categories. So the core genome, but all the genes found in all the strains in the species. Extensible genome or accessory genes are genes found in 2 or more strains of a species. And unique genes are genes specific to one strain among the species. And so just to give you an example of what's happening with E. coli that we study extensively in our lab. So back in 1997, the first E. coli genome sequence was published by Blattner et al. Now that was a laboratory strain. They found 4,288 genes in that particular strain of E. coli. Several years later, Pryor et al. Sequenced 2 isolates of E. coli, and they found additional genes that were not present in the first isolate of E. coli. But the core genes of those 2 strains of E. coli were 4,029 genes. So 4,029 genes were shared by these 2 isolates. But the pangenome was 5,416 genes. In 2008, 17 different isolates of E. coli were sequenced. And here you can see that the number of core genes decreased to 2,200. Only 2,200 genes were shared by these 17 isolates. So the most recent publication that I found was a group from China, where they looked at 491 isolates of E. coli, and only 167 genes were shared. But the pangenome size was 43,415 genes. That means that these 491 isolates of E. coli had more genes than all of the genes in the human genome. And so 867 represents only about 2% of all the genes in the pangenome. So how can we call these organisms E. coli if only 2% of the genes are shared? And so this is something that's really creating a havoc in bacterial classification, because what we thought we were considering E. coli probably belong to many distinct taxonomic groups. We call them E. coli because they share the same 16S ribosome RNA sequence, or the same biochemical characteristics. But at the whole genome level, they really are very distinct. And this is also confusing the way we think about infectious diseases caused by E. coli. As you know, E. coli is also found as part of the normal human intestinal commensal flora. But E. coli can cause all kinds of diarrheal diseases. E. coli can cause extraintestinal infections such as urinary tract infection, bloodstream infection, meningitis. So they're all called E. coli, but do they really belong to the same taxonomic organism? And so this is something that we're beginning to understand, that what we call pathogenic E. coli may actually be very distinct from the so-called commensal E. coli. So that was just looking at one genotype of E. coli called ST131. So ST131 is a very common, highly drug-resistant genotype of E. coli. They're all over the world. called UTI, urinary tract infection, bloodstream infections. So this group analyzed 4,071 isolates from different bioprojects from all over the world. And they found the core genes to contain only 3,712. So this is just a single genotype. Even within the single genotype, there's a lot of differences. So here, so-called core genes, those are the genes that were present in 95 to 99%. There were only 242. L genes are those genes that were present in 15 to 95% of the isolates. There was about 1,000. Genes are those that are present, shared by less than 15%. So most of the genes are actually unique genes. So even within the same genotype, there will be a lot of differences at the whole genome level, and then they probably belong. These organisms probably belong to different taxonomic groups. They should not be called E. coli species. Unfortunately, we're stuck with the term E. coli as a species designation. But with this new information, we may have to really change the way we consider what we consider to be species. And this applies not just to E. coli, but to many other bacterial organisms. So the pan-genome size of the ST131 was 26,000. So the core genes was only 3,712. Only 3,712 were shared among all of these 4,071. Now they were able to divide these 4,071 isolates into subclades, A, B, and C. And C happened to be the most common. So by further discriminating these strains, we can begin to understand the distribution of these different subgroups, and then understand what sort of diseases they cause and where they're distributed and how they're transmitted. So we can really begin to refine the sort of dynamics of transmission of what we call E. coli by having these additional information. In our lab, we've been studying another very common sequence type. It's called ST95. In fact, this particular genotype is the most common cause of urinary tract infection here in Berkeley, in the Berkeley campus among students. And one of the interesting, the reason we study this particular genotype is that despite the fact that this is the most common genotype, this organism, this particular genotype, rarely becomes multi-drug resistant. It might become resistant to one or maybe two drugs, but it rarely becomes multi-drug resistant. Whereas although the other major genotypes on campus are a huge proportion of those are multi-drug resistant. So we asked our question, is there some genetic features in this particular genotype that enable them to resist becoming resistant to antibiotics? And that was the question we asked. So we did whole genome sequence analysis of more than 1,700 ST95 strains. Then here we found the pan-genome size to be 35,000, more than 35,000, and only 10% of the genes were shared by all of these 1,749 isolates. The accessory genes was almost 90%, and less than 15% of the isolates belong to what's called cloud genes. And 40% were unique to only one isolate. And to make the long story short, we ultimately found several genes that were significantly associated with the pan-susceptible strains of ST95. And unfortunately, I don't have time to go into this, but we're able to find that one of these 15 genes was shared by yet another ST type called ST420, which also rarely becomes resistant. And this particular gene turns out to be one of the DNA repair polymerases. So you don't have time to go into this, but we're actually intrigued that this is a gene that is shared by these strains that don't become resistant, multi-drug resistant, and that somehow has to do with this particular type of polymerase is able to bypass DNA mutations, chromosomal DNA mutations. And so this allows these strains to resist the antibiotic stress that's created. So antibiotic stresses can cause DNA damage, and this polymerase can bypass those damages so the organism may continue to survive. Anyways, I'll tell you about this some. Okay. Similar types of studies have been done with other organisms like Klebsiella pneumoniae, Staphylococcus, Mycobacterium tuberculosis, and I just mentioned the ST131. So, as I mentioned earlier, the one major goal of molecular epidemiological investigation is to carry the subtyping of the organism beyond the species level. And so one way to do that, of course, is just as phenotypic methods, such as serotyping or drug resistance. But ultimately, we want to do genotyping and identify a clone. So, but then what is a clone? The standard definition of a clone is any isolate or group of isolates descending from a common precursor or strain by non-sexual reproduction. The loose definition is a group of bacterial strains belonging to a sample species that are indistinguishable by genotyping tests, such as multi-local sequence typing and PFGE, which I'll talk about next in the next session. These are some of the what I consider to be now conventional molecular genotyping tests. And so clonal complex is based on consensus definition used to include variants of genotype. So what happens when a whole genome sequences are compared? So these are the type of questions what people ask. What is the threshold or the number or type of nucleotide substitutions needed to exclude or include a group of bacterial strains in the same clonal group or complex? For instance, there's an E. coli, another common E. coli genotype, MLST genotype called ST69. If you do the PFGE on the ST69, they have nearly identical PFGE electrophoretic banding pattern, as you can see here. Okay, this is an AB. But if you do genome sequence analysis, there are some differences, as indicated by the red letters here. So these are nucleotide changes. Can you say that these are identical strains? Yes, they will be considered to be identical by PFGE, but certainly not by the sequencing method. So evolutionarily, they appear to be distinct, but they may be related as well, that maybe one descended from the other. Here's a situation where you have deletions. So in strain A, you have the GCG, as indicated in bold, that got lost in the strain B. So again, these could be potentially evolutionarily related, that the strain B may be a descendant of strain A. So here we can talk about evolutionary relatedness, but are they epidemiologically related? Well, you can't really answer that second question without additional information. But you have to, in order to answer the question about whether or not they're epidemiologically related, you really have to do epidemiological investigation. You can't just answer that question by looking at sequences or any genotyping methods. So evolutionary relatedness is based on a model or some sort of consensus definition. And you can use things like the number of single nucleotide polymorphism changes per genome over time. But epidemiological relatedness is not the same thing as evolutionary relatedness. This is it can be based on evolutionary relatedness data, but it has to be ultimately based on empirically validated data, meaning that it has to be done with some sort of epidemiological investigation, such as outbreak investigations or using surveillance data. And that's what ultimately tells you whether or not they're epidemiologically related. Okay. So you can also do some sort of analytical study that identify a risk factor associated with the clone or clonal complex. So if all of these different strains that appear to be evolutionary related share the same risk factor, then you can say that they're epidemiologically related. So you can use other types of empirically determined relatedness data, independent of genetic information. So this is a very important concept. This is one of the points that I also want to stress. So just to wrapping things up. So the scope of molecular epidemiology, the next generation molecular epidemiology, back in 2009, these were the different types of epidemiological issues or problems that were addressed by these molecular biology methods, such as tracking strains across time and geography. Yes, this is epidemiology. This is an epidemiological type of epidemiological investigation. But in my lab, we don't really do this that much because it's really not that interesting. Tracking strains across time and geography, you can talk about how, for instance, the so-called Latin American Mediterranean strain of Mycobacterium tuberculosis found in South America. And they can speculate that maybe they were brought to South America by the colonialists from Mediterranean countries, etc. And you can keep talking about this, but these types of studies have never saved a single life. And in epidemiology, in public health, what we want to do ultimately is to save lives and prevent morbidity. So these types of studies are interesting to maybe talk about over dinner or beer, but they're really not that interesting in terms of from the perspective of true real-world public health. And so what we need to do with molecular epidemiological investigations is to do things like distinguishing endemic from epidemic disease occurrence. It's a very important concept, and I think I will illustrate by using an example later on, maybe tomorrow, that as far as infectious diseases are concerned, there's no such thing as endemic disease occurrence. Every disease occurrence, infectious disease occurrence, is part of an epidemic. And stratification of data to define study designs. And I'll give you an example of this tomorrow again. Distinguishing pathovars from commensal. And I just alluded to this a few minutes ago by talking about E. coli. How do you distinguish pathogenic strains of E. coli from commensal E. coli? In studying microorganisms associated with hospital or institutional infections and identifying genetic basis for disease transmission. So these are some of the applications of molecular biology techniques to address epidemiological questions back in 2009. But because of the new technologies that we now have, we can do more. Yes, we can keep tracking strains across time and geography, distinguish endemic from epidemic occurrence, data, distinguishing pathovars from commensal, identify new modes of transmission. But in addition, we can we talk about healthcare and institutional thing. But in addition, we can also use these techniques to do surveillance and monitor response, analyzing population dynamics of parasitic pathogens. So many of the examples that I will use focus on bacteria because I happen to study just bacteria. But these technologies can also be applied for protozoan and helminthic organisms. Identifying genetic basis of disease transmission, I mentioned. Validate microdiversity phenotyping methods applied to epidemiology. Virus quasi-species population structure analysis. Identifying direction and chain of transmission. Conventional molecular biology until maybe 5 years ago could not do this, identifying direction and chain of transmission. So only because of the whole genome sequencing that we are now able to identify the direction and chain of transmission. Identifying hidden social networks and transmission links. Analyzing microbiomes to study non-infectious diseases. So these are what I call, these are types of studies indicated in bold what I consider to be next generation sequencing technology or next generation epidemiological investigations. So when do you use molecular biology techniques to assess an epidemiological problem? And just because a molecular technique is available, doesn't mean you have to use it. People like to just because, somehow it's considered to be better, or it's not necessarily any better. In the old days, the biology departments were called, microbiology departments were just called microbiology departments. But later on, they came to be called molecular microbiology departments. Why is that? Biology is microbiology. So when we use molecular microbiology techniques to do epidemiology, why do we call it molecular epidemiology? Have you ever heard of epidemiology when we use immunological techniques to do epidemiological investigations? Have you heard of biochemical epidemiology when we use biochemical tests to do epidemiological investigations? No. The reason this word, molecular epidemiology, came into existence because it just happens to be a very fancy term. When you use the word molecular, when you add the word molecular to something, it somehow sounds very cheeky and special. And if you write a grant, maybe you have a better chance of getting to use the word molecular somewhere. This is really the only reason why the word molecular is used in molecular biology. So there's no really, and truly, there's no such thing as molecular epidemiology. Molecular epidemiology is epidemiology. So when do you use molecular biology techniques? For not necessarily speed. Speed is sometimes important, as was it witnessed, of course, with COVID pandemic. But basically, we want simplicity, and we want high throughput, the capacity to process a large number of samples simultaneously. It has to be low cost, appropriateness. So these techniques have to have the capacity to address epidemiological questions that are not possible by conventional methods, and predictability. So the capacity to yield results consistent with the working hypothesis. The results must be both biologically and epidemiologically plausible. There are other criteria that we use for evaluating performance of strain typing techniques. One by the European group, Maslow et al., the European group. Typability, reproducibility, discriminatory power, ease of interpretation, ease of use. The European group was similar criteria. Stability is really the same as reproducibility. Reliability, epidemiological concordance, meaning that any technique that you use must be able to explain the epidemiological behavior of an infectious disease. So have some sort of concordance and typing system concordance. If you come up with a new genotyping technique, it has to also be validated against an existing, already validated. So one of the important things I always ask my students before they begin any sort of epidemiological investigation is to ask a question. When I go to Brazil and other countries, when I look at the students' thesis work or some research projects, they always ask me things like, what technique should I use to do the investigation? I genotyped 1,000 strains of Salmonella. What do I do with the data? And I asked them, so what was your hypothesis before you genotyped 1,000 isolates of Salmonella? What was your research question? And they often can't tell me. The most important thing I can say is before you start any sort of genotyping analysis, make sure you have a hypothesis or a research question. And so when I say questions, I don't mean things like which molecular typing method should I use, or how should I analyze the PFG patterns that I generate from my collection of isolates? What kind of information should I get to compare the strain typing data? What is the best genotyping method for Salmonella? These are not what I mean by questions. You can ask these questions, yes, okay, but this is not what I mean by questions. I mean by questions, the questions must be based on a hypothesis, a set of hypotheses. It's about methods. It's very important. Otherwise, you will never be able to do a good molecular epidemiological investigation. So these are some of the references that cover some of the points I made. So you can, I think this talk will be given to you. And so you can just look at the references and look at these references. So I will stop here and share so I can entertain questions.
Thank you, Dr. Lee, really, for a nice presentation. And it's open for discussion. The audience, here the participants. He already ended his talk by putting the questions. Now it's your challenge to ask questions now.
So if there are no questions, let me ask you questions for the participants. How many of you are microbiologists? Raise your hands if you're a microbiologist. No? How many of you are public health investigators or public health researchers, or you work with public health? Epidemiologists. How many of you are epidemiologists? Okay, good. Well, that's good. You know, I introduced you some of the laboratory concepts, and then my next talk, I'll go a little more detail about the laboratory methods, so you can be exposed to these basic microbiological techniques, and you hear the vocabulary used in microbiology. So in Brazil, what we do is something similar to, I guess, what you were doing at the end of this week. We usually have public health people, epidemiologists. About half of the participants, epidemiologists, are public health people, and about half are microbiologists. But we do the practicum workshop at the beginning of the workshop. So on Sunday, the public health people are exposed to PCR techniques. So they're introduced to the vocabulary used for doing PCR. And then the microbiologists undergo just a quick overview training in R, the program R, which is a data analysis management software. So they go through a quick one-day training in that. And so that introduces to the microbiologists the vocabulary used in epidemiology and public health. So by the time the workshop begins, everybody's sort of somewhat familiar with each other's vocabulary. It makes it easier for them to, I think, follow some of the discussions that follow. So maybe there's something that we could do also in the future in Mysore.
I have a question. This is Dr.
Okay.
Sumant, microbiologist. How is molecular epidemiology useful in tackling the antimicrobial resistance that's a global problem? I just wanted
that's something. That's something that we do in our lab. Antimicrobial resistance is a huge issue, as you know, in the world, and this is something that we've been studying for the last almost 15 years. We focus largely on gram-negative bacteria because I think that's where the real AMR issue is. Gram-positive, like staph, MRSA, it's urgent anymore because you know, we now have better antibiotics for MRSA, but with gram-negatives, drug-resistant gram-negatives, we really don't have good antibiotics. We're running out of good antibiotics, and the number of mechanisms of resistance among gram-negative bacteria are far greater than what we see with gram-positive organisms. And so it's a real challenge. We use molecular microbiology methods to sort of study the epidemiology of AMR gram-negative bacterial infections. And I'm going to talk about this tomorrow in more detail. I'll give you some examples of what we've been doing with urinary tract infection. So, but it's a very important issue.
Dr. Lee, I have a question. NGS introduce more variations, help us to know more variation among the isolates and these among the strains of bacteria. What are the possible causes for this other than the known causes? Any unique causes for this? The NGS help us to understand.
Unique causal. What? I didn't quite hear what you said.
What are the causes for the variations among the isolates and the strains of bacteria in the same species?
Oh, I see. So with an example of the E. coli that I gave you, number one, it's like, I guess, a sort of a historical accident, calling E. coli. And it's only because of the whole genome sequence technology that we now have that what we've been calling E. coli are really comprised of maybe many different types of taxonomic groups. And so
I think we're not able to listen to you.
Okay, you got muted. Okay. So what I was saying was that the E. coli, the name E. coli, really it got kind of stuck in microbiology because of just a historical accident that we happen to rely on 16S ribosome RNA sequence and biochemical tests to call E. coli, but with the whole genome sequence, we now know that what we call E. coli is comprised of many, many different taxonomic groups. But I think there's something common to these organisms that are called E. coli. And I think what is happening is that these organisms are constantly evolving. They're able to horizontally exchange genetic materials. And they do this in order to adapt to new ecological niches. And so they're commensal E. coli. They have all of the genetic materials needed to reside in the intestines of warm-blooded animals. But if the E. coli were to then cause UTI or bloodstream infections, it has to gain additional genes in order to be able to replicate in the blood or in the urine or in the bladder. And so then many of the E. coli strains can also contaminate food and survive in food products outside of the human body. And again, in order for this E. coli to be able to do that, it needs additional genes for to be able to replicate in the ecological niches. So what I try to look at when I look at these pathogens, everybody wants to identify new virulence genes. And I don't like the word virulence genes because that's a human-centric terminology. From the perspective of the microbes, they're not trying to cause disease in people or animals on purpose, right? They're just trying to survive and replicate in these different environments. So if an E. coli happens to cause urinary tract infection, from the perspective of the E. coli, it's only trying to replicate inside the bladder. Of course, that will end up causing disease in humans, but from the perspective of the E. coli, it's only trying to replicate and survive in the bladder. And so instead of calling these genes that allow them to replicate in the bladder virulence genes, we should call them niche adaptation genes. And this is how I think these microbes are able to spread and disseminate in different ecological niches. And if they just happen to infect humans and replicate in some sort of organ in humans, we call them disease, but from the perspective of this microbe, they're only trying to survive in those niches. So I don't know if I answered your question.
We can classify that spontaneous mutation, another induced mutation. The environment where the microbes are living, the different kinds of environment, even in the culture test or in a host system, that environment also can induce further adaptation and new variation.
Exactly.
The summary of what we explained that also.
Yeah,
Exactly. The microbes in the host or in the culture that also can induce, that's why you could see more variations by NGS. We can identify all these subtle variations, right?
Exactly. Yeah, we have this technology now that we can really begin to see what's really going on.
I have a question, Dr.
And that's epidemiology, right? If you try to study how these microbes adapt in different environments and then cause disease in humans and non-human animals, that's epidemiology.
Yeah, I have a question. The E. coli which are causing sepsis, to become septicemic, do they need to acquire extra genetic characteristics or genome?
Yes.
Yeah.
Yes, that's what we're proposing. We don't know what those genes are yet, but that's the kind of research that we do. And so we're trying to study the genes that are necessary for the E. coli to replicate in the bladder, in the urine, but also in the blood. And
Yes.
it's the sort of the bioinformatics analysis that's required to do this kind of analysis is very complex, but my students have access to supercomputers here on campus, and this is what they do. So they can really, if we have enough samples, enough whole genome sequences, we can actually do this kind of analysis and come up with some genetic features that are associated with those that are the strains that are associated with sepsis or urinary tract infection, etc.
Okay. Is it predictable by any chance? So some of the people who have some complicated UTI, who have E. coli as the pathogen, just analyzing the E. coli that is isolated, is it predictable that it can go into septicemia?
So that's ultimately the sort of the practical application of the knowledge that we might gain from doing this kind of analysis. So, if we do identify genetic features associated with sepsis, then we're going to do a simple PCR on the E. coli from, let's say, urine in somebody with UTI, and then say that this person is at high risk for progressing to sepsis. But we're still many years away from that, probably. But
okay,
that's the kind of thing we want to do.
Yeah, I mean, I can add to that. There are sequence analysis help us to understand there are hotspot, mutation or hotspot. Earlier, we used to say that mutations are very random. Now it's not so. And certain sequences are more prone for undergoing mutations. This NGS has helped us to understand that. So that will help us to predict that if these sequences are there, these are the very hot spot, we can think about that, and any genes associated with that or any of protein it produces, we can think about plan for that to protect that or even to make a resistance for that also. So there are mutation hotspots. The genome sequences help us to understand.
Exactly, right. And in addition to these hotspots, especially with organisms like E. coli, they can also undergo horizontal genetic exchange. So they can gain new genes from other bacterial species in the same environment. So as you know, with the intestine, there's all kinds of other microbes, bacterial microbes. And so E. coli can acquire genes from these other microbes. And if those acquisition of those genes are advantageous to the E. coli will keep them and maybe adapt to different environments.
Any questions from the participant, please? Yeah, please. Yes.
Dr. Ali, thank you for the very informative talk. But if you do the sequencing from the clinical sample, and if you isolate the organism and then do your sequencing, how often do you see any market variation in the sequences? And if you do see difference, which would you give more importance to?
So, we do see, again, with examples of E. coli, because that's what we study. So when we do the whole genome sequence analysis of E. coli from different clinical sites, sources, we do see a lot of mutations or a lot of differences, not just mutations, but accessory gene differences. So it's not just a mutation. It's not just the SNP differences, but actual distinct genes themselves. So as I give you an example, even within like one sequence type, like ST131 or ST95, if you do sequential sequence analysis of different isolates, even within the same community, we see a lot of differences in accessory genes. And I think these accessory genes are being acquired or being exchanged among themselves, or being acquired from other bacterial species in the environment that the E. coli is in. And so we happen to sequence those strains, we're catching them at the time maybe where they recently acquired these genes. We don't know for sure, when they might have acquired those genes, but
for example, in tuberculosis, if you directly sequence the TB bacilli from the sputum, and if you isolate the TB bacillus in the lab, so how often do we see major differences in the sequences seen in the culture?
So with TB, TB is actually very interesting. TB is apparently is a relatively evolutionarily recent, it's an organism that recently appeared in the human population. And so it doesn't have the kind of level of different pangenome difference that we see in organisms like E. coli. But we do see differences. And with TB, it's not so much the difference in accessory genes, but we see a lot of SNP differences. And so with TB, for instance, the drug resistance that the TB organism acquires is largely due to SNP changes, mutations, and not the acquisition of drug resistance genes from other sources. And so TB seems to be undergoing changes maybe a little more slowly than organisms like E. coli. And I think that's also because, as you know, the only reservoir of TB is the human body, right? There's no reservoir outside of the sort of natural reservoir outside of the human body. So it has a very limited ecological niche in which it replicates. And so maybe it doesn't need as many different genes as organisms like E. coli, but we do see differences.
Thank you.
Yeah, basically, E. coli is present as a commensal and a pathogen, while Mycobacterium tuberculosis is, if at all it's present, it's not a commensal. It'll be dormant for some time and then becomes pathogenic. So the kind of differences that we see in E. coli must be enormous compared to what we see with the Mycobacterium tuberculosis.
Exactly, exactly. Mm-hmm.
Any other questions from the participants? You can find that.
So we start again at 11 your time?
Yeah.
Okay, so it's actually 10:25 p.m. here. Oh. So maybe if you can give me 5-minute break, then I'll come back at 11 o'clock your time.
Yeah, thank you for your first talk, and it's a illuminating talk, and everybody is, seeing how the molecular epidemiology, phylogenetic relationships, and molecular evolutions, how the differences one can draw, and where we stand. We have to be very careful now with new terminologies. Okay, thank you for your nice talk. So we'll be back soon in 5 minutes.
I'll be back in 5 minutes. Thank you.
Thank you, Dr. Reli.
Thank you, Dr. M.N. Sumana and Dr. N.B. Ramachandra for chairing the session. I now request Dr. Akhila Prashant, Professor, Department of Biochemistry, JSS Medical College, to kindly hand over the memento and certificate as a token of appreciation to the chairpersons. We will now have a tea break for 10 minutes and join back. A kind information for all the participants. Please join to your WhatsApp group through the link sent through a registered email ID.
Okay, I'm back. Let me share my screen. Okay.
Hello, sir. Is Kalim here?
Oh, hi, Kalim. Hi, how are you?
I'm good, sir. How are you, sir?
Nice to hear your voice. So you're attending the session. Oh, very good. How are you doing?
Good, sir.
Good. So we should talk at some point about your project.
Dr. Lee, how are you?
Oh, hello. Hi.
Hi. How's the baby?
Oh, the baby's good. Yeah, the baby is almost 3 months.
It's a he or she?
It's a he. His name is Max.
Okay, good.
Yeah, he's doing well. He's asleep now.
Okay.
Fortunately, I'm able to do this talk without any crying in the background.
I was thinking you are talking from the lab.
No, I'm in my one of my rooms in the house.
Okay.
So how are you?
All good.
Yeah, so I just got your paper, so I'll take a look.
Yeah, I sent it yesterday.
Yeah, thank you. I haven't had a chance to look yet, but I will.
Okay, take your time.
Okay, good.
It would have been nice if you were here today.
Yeah, no, I wanted to go. I really wanted to go. I really wanted to be there. Maybe next year.
Next time, yeah. I think they will take another 10 minutes to get back inside.
There's a tea break now.
Okay. So I'll just mute myself and then.
Yeah, you can come back after 10 minutes. Okay, bye.
Sir? Oh,
आज कौन की गेस्ट आ गए थे हाँ पहले एक पोटा आया 15-20 साल का था 15-20 नहीं 14-15 साल का फर्स्ट आको जरा खत ट्रेडमिल के पास से खड़े हैं मैं क्या कि बताना शुरू करा मैं जाकर अंदर श्रीजीत को पूछ गया उन्हें अंदर जाकर श्रीजीत को पूछ को श्रीजीत उसे साइकिल मारने बिठा दिया था वो ऊपर इस वाला उन्हें होटल गेस्ट जरा खत में उसका बाबा आया बाबा कौन सी कहते ये करो क्रिकेटर बनना की क्या की बनना हो क्रिकेटर बनना चाहता है बाद में उसका बाबा म्हणजे मेरे तरफ उसे बोल रहा है सो या you are 14 years old and see this is 15 years young करको 50 years young करको म्हणजे बसाक बोलता है रनर मालूम नहीं श्रीजीत बोला था कि नहीं बोला रहा ना पहचान गया उसकी उम्र था उसने भी बाबा भी कम से कम वो उम्र का था
Okay, welcome back. We will again have a talk by Dr. Lee Riley. I invite Dr. B. Madhu, Professor, Community Medicine and Deputy Director, Research, JSS Medical College, JSS Academy of Higher Education and Research, and again I invite Dr. N.B. Ramachandra, UGC DSR Fellow, Department of Genetics and Genomics, University of Mysore, to chair this session. Thank you.
Thank you.
Thank you. Dr. Lee, you are around? Shall we start now?
Yes, I'm here. I'm ready. Okay.
Yeah, I think we can start now.
Okay. Good. Can everyone see my slide now? Slide is posted?
Yes.
Okay. Okay. So I'm going to quickly go over some basic laboratory methods used to do molecular epidemiological investigations. And of course, go over some conventional methods that have been used, and then go into some of the molecular biology methods. So just basic learning objectives here. Name the phenotypic microbiological methods used to subtype microbes for epidemiological investigation purposes. I'm going to talk about PCR-based genotyping methods applied in epidemiological investigations. And then three types of next-generation sequencing methods that can be applied to conduct epidemiological investigations. So investigations, of course, when we do these types of investigations, we're epidemiologists are interested in doing like outbreak investigations, surveillance, and then research. So these are three sort of major activities of epidemiological investigations people do. And the application of laboratory techniques to do investigations. First, you have to be able to diagnose the cause of an infectious disease, and then for the purpose of diagnosis, you identify the organism to the species level. And then you can do strain typing. And that's used to do further investigations, and especially epidemiological investigations. So diagnosis could involve just a simple microscopy to look at morphology on plate growth, tests such as MRI, PET scans, pathology, biochemical tests, serology, gene probe, PCR. Strain typing, we can do further biochemical tests, functional assays, serologic tests, and so when we say phenotypic methods, we're looking at observable characteristics expressed by a cell or an organism, such as drug resistance, virulence, or morphology. When we say genotypic methods, we're examining genetic characteristics of a cell or an organism according to its genome or specific genetic loci. And so conventional methods that are used to subtype organisms include examining growth and morphologic characteristics, biochemical characteristics, serology or immunological characteristics, functional and physiologic characteristics such as antibiotic susceptibility, phage typing, closing typing, cell culture assays, toxicity assays, in vitro survival characteristics. many of these techniques are no longer used widely, such as phage typing, Coulson typing, really something that's fallen by the wayside. Maybe reference labs might still do this, but because of, again, the technologies like the whole genome sequencing, it's no longer necessary to really do these types of conventional methods. Toxicity assays are still done in some labs, some reference labs. And the virulence assays are done in some research labs, but not routinely done in clinical labs. Multi-locus enzyme electrophoresis is one of the methods. It's an old method that's not widely used anymore for epidemiological studies. So taxonomic methods, growth in colony morphology, we sort of briefly mentioned before. You can classify bacteria into things like cocci or rods or spirochetes. You can also look at the way they grow on plates, such as the color that the organism produced, the shape of the colonies, the pattern of colonies, the biochemical characteristics that the colonies produce, kind of a mucoid type of pattern that some organisms might produce. There's a number of morphological structures that one can examine on colonies. Chemical characteristics, you can do these tube sugar tests, but these days, these kind of biochemical tests are done in these automated systems in most labs. Serologic tests are based on the serologic response to microbial antigens. So when we look at gram-negative organisms, we do look for antibodies against the flagellar antigen, which is called H antigen, and we also look at antibody response to the O antigen, which are the saccharide antigens on the bacterial surface. And so CDC would use these serotyping results to do surveillance. Here, in the case of Salmonella, serotyping has certain, they're designated with certain names, descriptive names such as Salmonella enteritidis, Salmonella typhimurium, Salmonella Newport. With E. coli, they're given numerical designations, such as E. coli O157, H7, which many of you know is a major cause of bloody diarrhea associated with outbreaks, foodborne disease outbreaks. But these are all really based on all H antigen, the antibodies that's elicited against the flagellar H antigens and O antigens, polysaccharide. With viruses such as influenza, again, they're based on the antibody response against the different components of the virus, gluten and neuraminidase enzymes, and they can be classified, and then they can be used to track the epidemics of these subtypes of the influenza virus, and also for surveillance purposes. So serotyping, so in the case of the influenza virus, it can be subtyped by serotyping or genotyping. Susceptibility tests, there are many ways of doing drug susceptibility tests. The most common and simple is the so-called disc diffusion test, where you put these discs that are impregnated with antibiotics and put it on a lawn of bacteria. They look for zone of inhibition, as you can see here on this slide. So if there's no zone of inhibition, that means that the organism is resistant to that antibiotic that's in that disc. The plate that's shown on the right is a type of a diffusion test. It's called the E-test, which enables one to see to actually quantitate the level, the concentration of the drug at which the organism is resistant. There are also broad dilution assays that people can do. Of course, these days, again, with the whole genome sequence analysis, and you can identify the specific drug resistance genes that are involved in conferring drug resistance. So that's sort of a quick overview of the conventional non-molecular biology methods used to subtype microbes. Any questions at this point? Okay, let me continue. So now we're going to talk about the molecular methods, molecular microbiology methods. So these molecular microbiology methods can be really classified into three different groups. Those that are based on the electrophoretic band pattern analysis. So these are fragments of DNA, pieces of DNA that are resolved by agarose gel electrophoresis, and you look at the patterns of these bands in a gel. There are also methods that rely on nucleic acid hybridization. So these are sort of the kind of microarray technology methods that people can use, but also southern blot hybridization. There are many techniques that are used. These methods are really, again, gradually being faded because, again, the sequencing technology is really becoming more affordable and also more easily analyzable with some of the tools that we have. So this third category is nucleic acid sequencing, and really much of the genotyping methods rely on this third category, the nucleic acid sequencing method. In the old days, people used to use plasmid profile analysis. When I was at CDC as an EIS officer, this is what we used. We crack open the bacteria and resolve the DNA. We remove the chromosomal DNA and then resolve the plasmids in agarose gel. And by looking at the pattern of these plasmids in the gel, we can infer strain relatedness. Genomic fingerprinting, there are many types, chromosomal digest, such as restriction fragment length polymorphism, RFLP, PFGE, you can do segmented RNA gel electrophoresis, ribosomal RNA gel electrophoresis, PCR-based methods, and then nucleic acid hybridization and nucleic acid sequencing. So as I mentioned, these fingerprinting methods are gradually being done away with. And in fact, CDC had a surveillance system that was based on PFGE until recently, but that's now largely being replaced by whole genome sequencing. So that's part of their foodborne disease surveillance system. So in the old days, genotyping techniques were classified into those that looked at microdiversity and macrodiversity. So microdiversity, these are techniques that were designed to analyze changes that occur in part of the genome of a microorganism. But these days, because of the whole genome sequencing technology, we can analyze the whole genome and then study the different parts because you have essentially the information in the whole genome. So I'm going to cover some of the old methods because these are still some used in some places and in some research settings, they're still used. So I'm just going to quickly go over some of these. So first talk about the electrophoretic band pattern analysis and then PCR-based methods and nucleic acid sequencing methods. So I'm going to give I'm going to use Mycobacterium tuberculosis as an example to illustrate some of these techniques. So with Mycobacterium tuberculosis, in the old days, the gold standard was what's called the IS6110 RFLP analysis. So IS6110 is an insertional sequence that's randomly distributed in the genome of the Mycobacterium tuberculosis. And so you essentially take isolate the DNA from the Mycobacterium tuberculosis and then chop up the chromosomal DNA with an enzyme. It's called PVU2 enzyme. And then you run these fragments in a gel. And then you take a probe and hybridize against the one fragment of the IS6110. So if a particular fragment of DNA contained the IS6110 fragment, it will hybridize to the probe, and then you can detect it in the gel. So that's called IS6110 RFLP. Very cumbersome, labor intensive, but that used to be the gold standard. Follicle typing is another technique that's used to type mycobacterium tuberculosis. And Miru VMTR, oligotyping, repetitive element PCR, rapid PCR, and PFG. So RFP again, PFG and RFP southern blot hybridization, ribosome RNA fragment, geoelectrophoresis or ribotyping. These are all and segmented RNA geoelectrophoresis. These are all methods used on electrophoric analysis of nucleic acid. These are methods. And all of these techniques were developed to overcome the problem of ease of interpretation of patterns of nucleic acid restriction fragments that are resolved by geoelectrophoresis. Okay, so when the technique relied on having to resolve DNA fragments in a gel, the results needed to be interpretable. So these above techniques were developed to do exactly that. So PFG is an example. This is still used in some places. So this is the technique that separates DNA up to 4 megalodon length. And the movement of these large pieces of DNA depends on the DNA plasticity in the electric field that's applied in different directions. And so separation of the DNA then depends on the DNA reorienting itself as it goes through the gel matrix, as the electric field orientation changes. But there were some obstacles that had to be overcome to do PFG. One is that the DNA fragments that are more than 25,000 bases are poorly resolved by agarose gel electrophoresis. And then there's also a tendency for these large pieces of DNA to undergo autodigestion by these enzymes, endonucleases that are used to cut the fragments. And so what they did to address the both of these were to use these enzymes in what's called the agarose gel plug. So you put the extracted DNA in an agarose gel itself, and then you apply the enzymes. An enzyme will diffuse into the agarose and then chop the DNA, and that prevents the DNA from undergoing autodigestion. And then that's resolved by the gel electrophoresis. So with conventional gel electrophoresis, the fragments go straight like this because the polarity is only applied in one direction. So you have minus and positive sign. With PFG, the polarity is applied in different directions. That allows the fragment, these large fragments, to go through the gel and produce a pattern of the agarose gel. And there's a criteria that's used to interpret these gel patterns of PFGE, and they came to be known as Tenover's criteria. So Fred Tenover was a staff member of the CDC. He is now with a biotech company, but he came up with this criteria. And so by looking at these patterns, if you just look at these patterns, A and A1, if you just eyeball them, they look quite different. But if you think about this, if you look at the red band in A, if there's a mutation that occur in that fragment, the red indicated by the red line, red band, then that might become subject to that particular fragment may be digestible by an enzyme that's used. So that particular band will separate into two fragments, which are shown here in red. So these two red bands represents the two fragments generated by cutting at one spot in this fragment. And so here you have the patterns. If you look, there's a one band difference here, two band difference here, and three bands. So there's a difference of three bands. But these three band difference is due to just one single mutational event. So that's considered to be closely related. Here you have two mutational events. So same thing here. You have an enzyme that will, if this particular fragment has a mutation, the enzyme will recognize that particular site and cut it into two fragments. So this band generates two fragments, and this fragment also generates two fragments. So now you have 1, 2, 3, 4, 5, 6 differences. And those 6 differences in band are due to just two mutational events. So these patterns, these two patterns are considered to be possibly related. If there are more than 7 band differences, or if there are more than 3 mutational events, 3 or more mutational events, or 7 or more band differences, they're considered to be different strains. So this is, of course, with the whole genome sequencing, they don't quite match these Tenever's criteria, but until the whole genome sequence technology came to be applied, this is the type of criteria that was used to interpret these band patterns. They also pointed out that in order to really do this, you always have to compare the patterns to some reference pattern. You can't just randomly compare patterns, and if they appear to be similar, you can't just say that. You can't just conclude that they're related. You have to compare them to some known reference strains. So these reference strains could include things like a strain obtained from some recognized epidemic or outbreak, or some common strain that's been identified in the surveillance system, or some well-documented newly introduced strain in a hospital or community setting. So if you have those reference strains, then you can make these types of interpretations. You can use Tenever's criteria. Any questions? So as I mentioned, there was a surveillance system at the CDC called PostNet. And in fact, there's actually an international PostNet. And so these were really maintained by the national reference labs in different countries. And they would compare these patterns across labs. But the problem was it was very often difficult to compare these patterns across different laboratories. And so they were very cumbersome to use. And over the last few years, these post-field gel electrophoresis reference labs are being largely replaced by whole genome sequencing surveillance systems. So just some PCR and sequence-based genotyping methods. PCR methods that are used to look for virulence determinants. And so here, you have to know the target sequence to amplify by PCR. And you can amplify a single amplified gene product. And so these are usually used to identify some specific type of virulence genes or maybe antibiotic resistance genes. You can also fractionate multiple PCR amplicons. And so here, you're essentially generating fingerprint patterns by cutting with some sort of enzyme a large PCR product. And there's several techniques like this, such as AFLP and RFLP with single PCR product. Here's a type of a virulence typing of diarrheogenic E. coli that causes diarrhea. So these were designed to amplify a particular virulence gene representing the type of diarrheogenic E. coli. And they were designed to amplify the targets to generate different molecular weights. Okay, so the bands here represent this virulence gene, the heat labile gene of enterotoxinic E. coli. And then the lanes 3 and 4 represent, it's called an EPA gene target, which are genes that are needed by entero-invasive E. coli to enter or invade cells, human cells. In lane 5 and 6 represent bundle-forming pili genes, which are representative of what's called enteropathogenic E. coli, or EPEX. And then lane 7 and 8, they also they target the heat-stable toxin of enterotoxinic E. coli. And so the PCR products that are designed to amplify heat labile, heat-stable toxins, identify ETEX, or enterotoxinic E. coli, and then ones that amplify the bundle-forming pili identify the EPEX, and the ones that target this EPA-H gene, which is a gene necessary for invasion, identify entero-invasive E. coli. So this is something that can be done very quickly to identify different classes of diarrhegenic E. coli. And they can also, there's all kinds of other PCR-based methods that generate fingerprint patterns, repetitive elements. They can do so-called ERIC sequence analysis. ERIC stands for enterobacterial repetitive element intergenic consent sequences. These are repetitive elements found mostly in gram-negative organisms. REP, which stands for repetitive extragenic palindromic sequences. Box sequences, these are also repetitive elements found mostly in gram-positive organisms, bacteria, gram-positive bacteria. IS elements, such as the IS6110, insertional sequence of Mycobacterium tuberculosis I mentioned, and PGRS, which are polymorphic GC-rich repetitive sequences that are also found in Mycobacterium tuberculosis. And then the DRE PCR, or double repetitive element PCR, ITS PCR. So these are all the different PCR methods. The basic principles behind all of these is that you have these repetitive elements as indicated by the orange boxes. And you make primers that are directed away from these repetitive elements. So the spaces between these repetitive elements get amplified, right? And so these two primers here will generate this fragment, that primer, and then the other primer there will generate that large fragment, etc. So all these different fragments are generated, and you can then resolve these fragments in a gel, and that will produce a fingerprint pattern. So ERIC PCR, so not all gram-negative organisms have this ERIC sequence. So these are the ones indicated in black have ERIC sequences. So you can genotype these gram-negative organisms by ERIC PCR. The advantage of ERIC PCR is very quick, very simple, very inexpensive, and very fast. So when we don't have lots of money to do the whole genome sequencing, or we're in a hurry to subtype these gram-negative organisms, we do the ERIC PCR. And then we can quickly say whether these strains are related or not. So here's an example of ERIC PCR that was done on E. coli isolates from urinary tract infections. This was done by one of my former students, Amy Mangus, who's now a professor at the University of British Columbia. So she took all these E. coli isolates from the urine samples from students with UTI and just did this quick analysis. And you can see some of these have very similar patterns, such as, I don't know if you can see my arrow, but the two lanes on the left side of this gel. And then there's also you see some similar ones in the other parts of this figure. So this analysis led Amy to come up with a very interesting idea about the mode of transmission of UTI, which I'll talk about tomorrow. This is an example of box PCR analysis that was done on pneumococcal isolates, and this was done in Brazil by my colleagues, my research colleagues in Brazil. And Albert Koh, who is a professor at Yale, who was in Brazil doing this work at that time. And this is, again, it was a very quick way to genotype pneumococcal isolates. So you didn't have to do a whole genome sequencing, it's very cheap. And this was looking at the non-susceptible strains of pneumococcus that was non-susceptible to penicillin, and then those susceptible to penicillin. And they found in the study that the non-susceptible penicillin, non-susceptible strains of pneumococcus shared identical patterns. So the non-susceptible, penicillin non-susceptible pneumococcal strains causing meningitis in the state of Bahia, Brazil, was really a clonal disease. This is a very important finding that they made by this simple molecular microbiology analysis. VNTR stands for variation in the number of tandem repeats. So this is something that's found in a variety of bacterial organisms. So you can just amplify these loci, and they'll produce DNA fragment whose length vary between the strains because there are different copy numbers of these VMTR elements. And so this is something that's used in Mycobacterium tuberculosis. VNTR, MLVA stands for multilocus variable. I'm sorry, I can't remember the full name of the MLVA, but this is something that's used in Mycobacterium tuberculosis. Yeah, so here, Mycobacterium interspersed repeat units, MIRU. So this is what's used in Mycobacterium tuberculosis. So Mycobacterium tuberculosis contains about 41 loci with these direct repeats. So these a number of repeats can vary between strains, and so PCR could be done to amplify these segments, and they generate different sizes of these repeat elements. So this is the location of these MIRU elements in the Mycobacterium tuberculosis chromosome. So they're randomly scattered across the genome, and we can amplify these segments. And then the strain typing methods used nucleic acid sequencing. We've been talking a lot about the whole genome sequences already, and I think Dr. Carrie Cooper will be really going into more detail later this week. So he can tell you more about this. The old method, which is something that still applied in some settings, but this has been largely replaced by more the new generation methods. The old methods called the Sanger, chain termination methods. But once you have the whole genome sequence data, then you can compare subsegments of the genome. And there are many techniques for those. Multi-locus sequencing. You can do multi-locus sequence typing without the whole genome sequence information, but these days, because of the ready availability of the whole genome sequences, you can actually do MLST based on the whole genome sequence data. You can also do single nucleotide polymorphism analysis, microsatellite analysis. You can also compare extrachromosomal DNA elements, especially those that are found in parasitic organisms like protozoans and helminths, like mitochondria and kinetoplasts. Plasmids are found in bacteria. So let me talk a little bit about MLST, multi-locus sequence typing, because this is still something that's widely used. So here, you compare allelic differences among selected set of gene segments. And usually these gene segments, targets, are about less than 500 base pairs in length. And each set of allele sequences comprise a sequence type, which can be compared across different labs. And so with the example of a Staphylococcus aureus, these are the genes that are targeted. It's usually 7 genes that are targeted. And each of the genes has anywhere from 18 to 33 alleles. So the combination of all these different alleles will comprise a lot of different subtypes. So the steps are, you first do the PCR amplification of the target sequences using the MLST database protocol. This is a standard protocol that everybody follows. And then these are the amplified products are sequenced. And then you have to make sure that the consensus sequence used to designate alleles, L types, is accurate. And then you submit the sequence data to this MLST database site, which is maintained in the UK. And then there's a curator at the site, which will then send you the ST designation. Now, these days, this particular site requires whole genome sequence submission, and then they will send you the MLST designation based on the whole genome sequence gene sequences. And so the SNP analysis can be performed by concatenating all the allelic sequences. So concatenate means you create these false sequences by stringing along these different sequences of the amplified products. And then these concatenated sequences can be aligned to create a dendrogram, which is this sort of a tree that can create it. And the group assignments made by the dendrogram can then be compared to ST designations. So here's an example. So these are the amplified products, okay, and then they're sequenced, and then they're concatenated like this. And then you compare the different concatenated sequences to make a dendrogram. Okay, so you align this and make a tree. And this is how you do the SNP analysis. Okay, so that's pretty much just a quick overview of these techniques. And as I mentioned, Dr. Cooper will go into more of the next generation sequencing technology, which is really what's now largely used to do epidemiological investigations. So I will stop here. So this is just a quick overview. Any questions?
Thank you. It's open for discussions. Any questions, clarifications? This talk, even morning, first talk also, you can, if you have any doubts, you can clarify.
So these two talks were really done to kind of introduce the basic concepts and some basic vocabulary, because tomorrow I'll be talking about the application of these technologies to address very important epidemiological issues. And I'm going to give one example of salmonellosis, how some of these early technologies were used to understand the epidemiology of salmonellosis, and then some of the application of the new technologies to address the issue of urinary tract infection. And so I think you'll probably be able to understand these basic concepts by these specific examples of the applications tomorrow. So if there's no further questions, I think I'm going to sleep. It's 11, almost 11:20 p.m.
Here, I'm Dr. Madhu. I'm from the public health background. So as I am addressing the public health issue on how to control tuberculosis, we look at the population. The major concerns are adherence to treatment and drug resistance. How these kind of molecular epidemiological studies can have an insight or provide evidence to address these issues?
Yes, molecular epidemiology is very important for TB control. Unfortunately, I don't have time this week to talk about TB, but there are many studies that have been done. So, in my first talk, I sort of alluded to the importance of genotyping TB to differentiate reactivation disease from recent infection TB, right? And as I mentioned, that this distinction is very important because if most of the new cases of TB right now are due to recent transmission, that means that the TB control program is not working. So you have to do something about the TB control program. You have to ask yourself, why is the TB control program not working? So to really address that, you need to know what proportion of the new TB cases in your community are due to recent transmission. And so molecular microbiology technique can be applied to answer that question. And then more recently, these sequencing technologies are being applied to understand the transmission dynamics of multi-drug resistant strains of Mycobacterium tuberculosis. And again, this is very important because one can really track, by using whole genome sequence analysis, you can track the direction of transmission and the chain of transmission. Before the whole genome sequencing technology, we couldn't really distinguish who infected whom. If two people develop TB around the same time, you can't really tell for sure which one gave the infection to the other person. Okay. But with the whole genome sequencing technology, we can now do that. Okay. So there are many good applications of these technologies for TB control.
Yeah, when it comes to the sample required for these kind of analysis, and the sequencing is a costlier affair. So the sample size required when these kind of studies have to be done. So share about something on that.
Right. So yes, you're right that in order to really be able to make some sort of epidemiological conclusions with high confidence, you need to have large number of samples. But when you have to analyze large number of samples, of course, whole genome sequencing can get expensive, and so you have to kind of balance how much you can afford to do the whole genome sequencing, and then the number of samples you can get. And so that's still a limitation in applying the whole genome sequencing technology, but the prices are gradually coming down, and so I think in the near future, I think we'll be able to do this with more ease and affordability. So I mentioned Dr. Ranjita Gowda, who's in the audience now, was actually in my lab learning how to do the bioinformatics analysis of Mycobacterium tuberculosis. And she's applying this knowledge to look at the TB cases that occur in people with diabetes and people without diabetes. And we're trying to understand if there's something, some characteristics of TB strains that cause TB in people with diabetes and people without diabetes. And so she just finished writing her paper, and I'm going to be reading it shortly. So that's another type of application to address a public health issue, which is a huge problem in places like India, in Brazil. In Brazil, for a period of about 10 years, TB incidence was decreasing from about the 1990s to mid 2000s. TB incidence was really decreasing, but then the prevalence of diabetes started increasing, and this decrease in TB reversed itself because of the diabetes. So now the TB is rising largely because of the increasing prevalence of diabetes. And the same thing is happening in India.
Exactly.
That has a huge diabetes problem, and this is really contributing to the TB problem in India. So it's a very important issue to look at.
Can I add to one point for this? In India, we need not go for direct sequencing. We can use molecular markers like multiple drug resistance, extreme drug, drug resistance. We have first generation drug for TB, second generation drug. There are markers already available. In particular locality, we can use some markers. We can identify is it multiple drug resistant, extreme. Then only if need to be, we can go for sequencing. So markers available now. One had to go.
No, you're right.
Use
you're correct.
The second,
if you're interested in drug resistance, you don't have to do whole genome sequencing. You can just go after the drug resistance genes themselves. And there are many simple diagnostic tests that you can use to do that. And so there's a PCR-based methods. There's all kinds of simple tests you can use to rapidly detect the drug resistance genes.
Most of the PCR-based, I think can even one PCR run, if 500 rupees it will cost if you have the right kind of primers, that analysis can be done now for one sample. If you have multiple samples, even the cost can be reduced. Second point, that now the people are coming up in Bangalore, sequencing rates for whole human exome sequencing 14,000, including. And for this kind of sequencing, I think that's also secure. If need to be, we can go for it because the Illumina Neo machines have come, and it's a very cost-effective now.
You're correct. Right. You don't have to always do whole genome sequencing to
there are plenty of markers available, so you can do that. Proper diagnosis.
But if you have to really track the transmission, then you will have to do the whole genome sequencing. So it really depends on what your questions are. If your questions are addressing drug resistance, you don't need to do whole genome sequencing. But if your questions are really trying to understand the chain of transmission, you will need to do whole genome sequencing.
Okay. Thank you.
Sir, I have a question.
Is there any or much difference between studying viral epidemiology and bacterial epidemiology?
No, not really. I think, they have all these different terms. They're all epidemiology. So you can call them anything you want, but they all basically comes down to epidemiology. All of the analytical methods that's used to analyze the data generated from these molecular microbiology techniques are really the same. They're basic epidemiological methods. So the only difference is that you're analyzing data that are generated by molecular microbiology methods. But it's no different from analyzing, data that's based on what you can observe, such as, eye colors, hair colors, skin colors, etc. And so, you just happen to be using a more refined analytical method to generate these types of data. But the analysis comes down to really all of the basic epidemiological methods.
Thank you, sir.
Good. Any other questions? Dear participants, all the techniques what is listed, one should know the principle behind that and how they work. If you know those organization of the whole genome, the bacterial genome, and how these techniques work, and I think it's very easy to go further to understand more about the epidemiology or even treatment or all kind of applications, right? So it's listed that because these techniques are developed from last 40 years, and you should, if you need, you should go and search in Google, wherever you get our PDF files, you get these techniques and get knowledge more about that if at all, if you need it. But they may not apply now, but if you know the principle behind that, you can understand more about the organization of the genome and where exactly the problems in these microbes are there and in applications wise. So thank you very much, Dr. Lee, for giving wonderful talk, the overview, foundation talk, and I hope all the participants enjoy your talk, and thank you very much for your talk.
Okay, thank you. Okay, so I'll see you tomorrow again. Okay. Good night. Oh, good morning for you.
Thank you.
Thank you.
I thank Dr. B. Madhu and Dr. N.B. Ramachandra for chairing the session. I kindly request Dr. Akhila Prashant, Professor, Department of Biochemistry, to hand over the memento as a token of appreciation. Lunch has been organized in the guest house. I request everybody to join by 1 p.m.
Good afternoon all. We'll now begin with our second session on basic epidemiological concepts. I invite Dr. Praveen Kulkarni, Associate Professor, Community Medicine, JSS Medical College, and Dr. Kiranmayi Muralidhar, Junior Research Physician, Public Health Research Institute of India, Mysore, to chair the session on study design overview and epidemiological methods. I request the chairpersons to introduce the speaker, Dr. Purnima Madhivanan, to the audience.
Thank you and good afternoon, everyone. It gives me immense pleasure today to introduce Dr. Purnima Madhivanan. Dr. Madhivanan is an associate professor in health promotion sciences at the University of Arizona, an infectious disease epidemiologist by training. She completed her medical schooling from Mysore Medical College and has an MPH and PhD in epidemiology from the University of California, Berkeley. She is the founder trustee of the Public Health Research Institute of India and is director of the Global Health Equity Scholars Training Program in collaboration with universities such as Stanford, Yale, and University of California. She established a clinic in Mysore in 2005 while completing her PhD dissertation. This clinic, now the Prerna's Women's Health Initiative, has delivered low-cost, high-quality reproductive health services to over 24,000 low-income women living in Mysore district.
Thank you.
This site has been recognized as a scientific and industrial research organization by the Government of India and a training site for global health. Dr. Madhivanan serves as an advisor to a number of state departments of public health in the US, nonprofit as well as governmental research organizations in the US and in India. She is also a mentor to numerous students and fellows globally. Ma'am's global health work has brought unique opportunities for collaboration, such as the workshop we are having today. We welcome you, ma'am, and look forward to listening to you and learning from your talks. So the first talk that Dr. Madhivanan is presenting focuses on study designs. We all know that choosing the right study design is crucial in planning a research study. And this presentation will go through various study designs that address different research questions, how they may be classified, their advantages and disadvantages, and how they can affect the success and failure of a study. Over to you, ma'am.
Thank you, Kiran. Good afternoon, everybody. Thank you all for coming back. I was really worried that when you heard study design, epidemiology, it's connected to humans, you work with organisms, so maybe not of interest to you. But I'll make it interesting, and you will like it. So, what I want to do with this presentation is give you the bare minimum overview. And if you're still excited, and if you're still interested, I will leave my email address. And you can contact me and say, hey, we want to learn more. And I'm going to be back in June. And I'll come back and give you multiple lectures that maybe some of you may go to sleep. But for this one, I will just try to make sure that you are interested enough to decide whether you want another lecture on study designs or not. Okay, so what we're going to do today is look at all the different study designs in epidemiology. What are the advantages? What are the disadvantages of using each of these study designs? And when do you use a certain study design? That's what we're going to do right now. And I don't know if we have enough time to go through sampling. We've already started pretty late. I want to at least get to the top main pieces about all the different study designs, and then maybe have a little bit of a discussion. So when we talk about study designs and epidemiology, what we're talking about is population health, trying to understand patterns in population health as to why do you see some groups having a disease and some groups not getting the disease in spite of being exposed to a certain risk factor. So when you think about study designs, you have to have three main concepts clearly in your head. And without that, I was just talking to one of you and I asked a very simple thing. What is your exposure? What is your outcome? And without clearly defining those two main aspects, you cannot do epidemiology. So the basic tenets of epidemiology is to answer the question, does an exposure cause an outcome? And what are all the other factors we have to keep in mind to make sure that we are not mixing up the effect of what you think is an exposure is happening because of some other covariate, which we sometimes can call as confounders. So you have to think in terms of your own study question, what is the exposure? What is your outcome? And what are all the different variables that can be related either to your exposure or to your outcome or to both? That's the first step. I've heard so many of you come and tell me, I have all this data, I have all of this stuff, but I don't know what my question is. So I'm asking you to flip it. Forget about what data you have. Doesn't matter. What you need to first clearly write out in your head is, what is your study question? And that study question will tell you what is your exposure and what is your outcome. Without that, there's no point in me even having this discussion on study designs, because all the study designs are trying to do is to examine that relationship between an exposure and an outcome. Okay? So when we talk about exposures and outcomes, as I said, the main reason, the main goal of doing epidemiologic research is to understand causation. Causation is a loaded term. We epidemiologists, we are afraid to use the word causation or causal, because the minute we say something causes something, it's a whole amount of new science where we have to ensure that there is no other factors mixing up the effect. And invariably, we think that this exposure is causing this outcome, but it's actually the third variable that's doing all the work. So we have to be very careful when we use the word causation. So I am very careful. I don't use the word causation at all, unless I'm really testing a causal model. I usually say, is it associated? Is there a relationship? So you start there. Forget about causal. If you can find new causal pathways, you'll get a Nobel Prize now. We're beyond that right now. Let's start with a relationship and an association. So epidemiologic research is to examine the patterns of disease occurrence and understanding if there is a causal relationship between that exposure and an outcome. So for us to have an epidemiologic assessment, as I said, you need to have your exposure very clearly defined, your outcome very clearly defined, and all these covariates. They could be a mediator, they could be a moderator, they could be a confounder. All of them have different properties. That's why you always collect all this additional data when you're doing your studies, right? That is, again, you're trying to think, does gender in some way mix up the effect between my exposure and outcome? And that you might think is possibly a confounder. So when you start saying gender is a confounder in my relationship between an exposure and an outcome, then the next thing you will think about is, what kind of test will I use to find out whether it is a confounder? Then I will start saying, what kind of a variable is this? Is it a continuous variable? Like when you say your blood pressure is a continuous variable. Gender is a categorical variable. It's a yes-no variable. Male, female, yes, no. So based on how you define your outcome, then you will decide what test you're going to do to assess the relationship between your exposure and your outcome. So it's all connected. So what we're doing is going back to the basics. Know your exposure, know your outcome, and figure out, based on the literature, based on your own understanding, what are all the potential covariate information you have to collect so that when it comes time to test your hypothesis and decide what statistical test you're going to employ, you will have the information. Okay? So once you have all that, that's when you go ahead and think about the test. Now, we said your outcome is a yes-no variable. So it's a categorical variable. That means I am going to do a logistic regression. I'm not going to do a linear regression because my outcome is not continuous. I will do a logistic regression because my outcome is categorical. And that's how you will now quantify the association between your exposure and an outcome. So what do we mean by quantify? You will come up with a measure of association. A measure of association, when you do a logistic regression, is an odds ratio. Now you have a number. Then the next question is, how do you interpret that number in English? What does an odds ratio of 3.2 mean? That's another topic altogether. But are you with me so far? You got it? Good. So, as we said, depending on what your study question is, we will decide what the study design is going to be. You cannot come up with a study design without knowing what your study question is. You cannot come up with your study question if you don't have a clear idea what your exposure and your outcomes are. It's all connected, okay? So the next thing you're going to do, once you have your exposure, your outcome, and your covariates, is to figure out what is your research question. And your research question should have your exposure and your outcome clearly in that statement. Because if somebody comes and says, I need to talk to you about my study, the first thing I'm going to ask you is, what's your research question? And the next thing I'm going to ask you is, okay, so if this is your research question, what is your hypothesis you're trying to test? Because somebody said, I have all this data, I don't know what to do. I don't know if I have to retest some of my samples. My question was, what is your hypothesis? Because that can tell me if I need to tell you that you need to retest or add more samples to your database. Without having a research question and hypothesis, I cannot even tell you what study design you're going to use. Your study design is connected to how you ask your question. And based on your study design, you will use certain analytical procedures to test your hypothesis. That's why it's all connected. So for all of you, whether you're in your first year or in your final year of your PhDs, write out your research question. Write out your hypothesis. And based on that, when you come and tell me what should I do next, I can tell you what you can do next. Without that, I cannot help you. I've had so many of you come and ask me these questions, and I have the same answer for you. Without your research question, without your hypothesis, I cannot help you because I don't know. I don't know how can I even tell you what study design you can use or what statistical test you can use. Because you can use a wrong statistical test because some paper said, do a t-test. So you will go blindly go and do a t-test. But your outcome was categorical. Would you do a t-test for a categorical variable? You will not. But you'll come up with some number and you'll say, oh my God, I found an amazing result. And then you'll go to the journal. And then the journal will throw it back at you. Or you'll get a nasty reviewer who'll get really nasty with you and he'll make you feel like shit. If you don't want to be in that situation, it's very important that we first self-correct by ensuring that we know what we are doing before we send it out. Okay? So, when we talk about population health, Like Dr. Riley said, you can call it molecular epidemiology, biochemical epidemiology, whatever epidemiology you want to call it. Epidemiology is about people. It's about humans. It's about diseases, patterns, risk factors related to diseases in humans. So we are talking about study designs means we are talking about testing some kind of a hypothesis in humans. So when you have to decide, based on your research question and hypothesis, what study design you want to use, the first question you want to think about is, are you assigning an exposure to somebody? Are you telling some person, you take the drug, you don't take the drug? You get the vaccine, you don't get the vaccine. Are you manipulating the exposure? That's the first question you ask. Yes, no. If you are manipulating it, right? You're giving the drug to some people and you're not giving the drug to some people. And then you're going to go and check, did they get better or did they get worse? That means you're manipulating the exposure. In that situation, we call those experimental studies. Okay? So now the question is, you are manipulating the exposure. So it's an experimental study. Now, how you're assigning the exposure to a person. I'm going to say, I like you, you're a woman, so I'm going to give you the vaccine. You're a guy, I won't give you the vaccine. Am I doing that? Or I have a box with everybody's names in it. I'm just going to pick, okay, this person who is with me is going to get the vaccine, so I give the vaccine. Next thing I pick, that says vaccine, so the next person gets the vaccine. The next thing I pick, it says no vaccine, so the third person doesn't get a vaccine. That means I don't have control over who I am assigning the exposure. And it's happening by chance, right? Because I'm just picking it, whatever comes in my hand. So I am randomizing people to either get an exposure or not get an exposure. That means now I'm doing a randomized trial. If I am not doing that, and I'm deciding who gets what, then those are non-randomized trials. Okay? Good so far? Okay. So that's the experimental part, because we were assigning the exposure. But most times, we don't do that. We are not assigning the exposures. We are merely observing. Oh, okay, this person had COVID. Now, did this person get long COVID? Or this person was exposed to SARS-CoV-2. Now, did that person die? You're not going and saying, I'm going to give SARS to this person. You're just observing this person had SARS-CoV. Now, will that person get better or won't get better? So you're just merely observing. That means we are not assigning exposures. In that situation, all those studies come under observational studies, because you're not manipulating anything. You're just observing whatever is happening. So those are observational studies, and most of us are going to be involved in observational studies. Now, epidemiology, they make fun of us. What they tell us is epidemiology is all about denominators. Because you tell us, I have 1,000 samples. I say, I don't care. What I want to know is, what's your denominator? You got 1,000 samples out of 1,500 people? Or did you get your 1,000 samples out of 10,000 people? Because it's a very different story. So we are about denominators. And when we talk about study designs, the first thing we're going to ask you, when you say, I found this amazing association between exposure and outcome. I'll say, as compared to what? You have to have a comparison group. Because you can say, I found this amazing association. This drug worked so well in this group. And I'll say, as compared to which other group? Maybe it will work just as well in some other group. I don't know. If you say it works so well in your group, that means you have to have a comparison, right? So, study designs, when you think about it, you have to ask, does it have a comparison group or not? If there is no comparison group, all you're trying to do is, here is a whole group of people. I just want to know how many of them are feeling sleepy. That's it. Not asking anything else. That's a descriptive study. You have no comparison because you're not saying, as compared to all the people in this room, what about somebody in the other boardroom? Then you have a comparison group. Right now, you have no comparison group. So that becomes a descriptive study when you don't have a comparison group. If you have a comparison group, then you ask the next question. Is it an analytical design now? Because now you have a comparison group. Now you're saying this group versus this group. Now the question is, in which direction are you going? Are you starting with an exposure, going forward in time to find out what outcome they got? Or are you starting with a disease and then going back in time to find out what exposures they had? Is it forward or backward? But all of them have the same thing. They're trying to examine the relationship between an exposure and an outcome. Okay? So if the direction is from exposure to an outcome, like you said, somebody was exposed to Agent Orange in the war. Now you want to see how many of them landed up getting a cancer. So you're going to catch hold of all these people who went to war, who were exposed to this particular chemical, and you're following them in time to find out what disease they got. Now that is a cohort study. On the other hand, I was talking to somebody, she said, we have a lot of breast cancer cases here. Breast cancer is an outcome. Now the question is, why is it that you have so many breast cancer cases here? Now you want to find out, are they potentially exposed to certain things that increase their risk of getting the cancer? So you're going to go back to find out what exposures they had. In that situation, it is a case control study. And to me, case control studies are the most difficult studies to do. The most complicated study design is a case control study. I won't go into that right now. But at the same time, you're not going backward, you're not going forward, you are capturing the exposure and outcome information at the same time. Then it's a cross-sectional study. So all of these three are analytical study designs. So let's go through each of them. Many times you hear the word retrospective, prospective. And they say, oh, I did a retrospective study. Doesn't mean anything when you say somebody has done a retrospective study. You can have a retrospective study if it was a cohort design. All case control studies are retrospective. Doesn't tell you anything. Same thing for prospective. A clinical trial is prospective and a cohort study is prospective. Doesn't tell you anything. So it's a wrong way to talk about study designs. So don't use the words retrospective or prospective studies when you're writing your papers, because it doesn't give us any information. You really need to separate out the direction of how the exposure and how the outcome information were collected and how your study participants were recruited into your study. In your methods section, those two things have to be very clearly laid out. Because when we are judging your results or trying to interpret your results, it matters. When you say you had an odds ratio, or let's say you said you have a risk ratio of 3.2, but you say your study design was case control study, then I have to start thinking about it. How did this person get a risk ratio from a case control study? So it's very important that you very clearly explain the direction and how you recruited your study participants. And also, I've already told you, using follow-up study, longitudinal studies don't mean anything. Because based on the previous graph I showed you, cohort studies and RCTs are both longitudinal. They both have follow-ups. So it's not going to help anybody understand whether you did an experimental study or an observational study. The other thing we have to also think about is epidemiology is about populations. So we need to talk about the difference between a cohort and population. When we talk about population, it's an aggregate of people. And how you define your population depends on what your question is and what your geographical spread is. So there are a lot of different ways you can decide on how you define your population. And at the same time, there are two ways you can define it. You can define your population based on a specific event that happened. Like for all of us who have faced this pandemic, we are all collectively a cohort or a population of people who have been exposed to an event, right? So we are all connected as one study group because we all have a common event that we were exposed to. And when you think about a time, if we said 2020, everyone who was alive who were exposed to the pandemic, right? You can't suddenly have somebody born today become part of that group, right? It's a closed group. That's it. Whoever was in 2020 alive at the point in time when they announced the pandemic had started, that was it. That's a closed group. You can't have any new people coming into that group. So that's called a closed group, or that's called a closed cohort. You're a specific group of people where you won't allow anybody else to come in because you all had a common event that made you connected. Okay? And the second one is you can have an open cohort, which is people are coming, people are going by dying, people are coming by being born into this community. So that's the population. That's open. People coming and going. Okay? So when it is closed, it's a cohort. When it is open, we call it a population. So in terms of the hierarchy of studies that you can do based on the questions you have, the lowest, the poorest quality study design you can choose is a case report, right there at the bottom. That's the poorest study design you will ever have. And many people get excited. I wrote so many case reports. So what? Doesn't matter. Because that's the lowest quality paper you can write. But it's a paper. We all have to start somewhere. So if you have a really interesting case to talk about, write that case report. But just keep writing case reports when you have other valuable data is a waste of time. Because it means nothing. You're simply wasting people's time. You're wasting your own time. So if you have a unique case, maybe it was the first case of COVID in your hospital, and it was very unique. You write it as a case report, and that's a valuable one. Usually that's when case reports are valuable, when there is something very novel and something very new that has not been found and you just happen to find it and you're sure that this is not just by chance that you've figured this out, you write a case report. Then you start seeing that there are more people coming with similar presentations. Now, instead of just one case report, you have a group of 7 people with the same situation. Now, you have a case series. Okay? Again, not very, we don't care unless it's really novel and innovative. You move on. Then the next one is you have, here's an example before I go to the next one. Here's a case report. This was in 1985. We were just finding out about this virus called HIV. And it was found in the adults, but we didn't know enough about it in the pediatric population. So this was one of the first cases that was very clearly described in the literature on the first child who was infected with HIV. So here's the case report of the first pediatric case of AIDS observed in Switzerland. It was a 3-year-old African Swiss patient, most probably vertically infected from this asymptomatic HIV antibody positive Zairian mother. That means the mom had the virus, but did not have the symptoms, was pregnant, and passed the infection to the child. And that's what they're saying. The most probable vertically infected infection from an asymptomatic HIV antibody positive Zairian mother. Clinical symptomatology started at 14 months of age, and diagnosis was made at 22 months when medical care and comprehensive investigations were initiated at the clinic. So that was the first description of a pediatric AIDS case. That's why it's a case report. So case report, case series, then the next most easiest and common, most common study design is cross-sectional studies. These are very quick to conduct, and they are the least expensive studies that you can do. And what happens here is you are collecting the exposure information and the outcome information at the same time. And it's like a snapshot. It's like taking a picture. So at one point in time, you're asking them, do you smoke? Do you have any lung conditions? Do you have COPD? Were you diagnosed with lung cancer? Everything we're asking at the same time to that person. So exposure and outcome information collected at the same point in time. It's usually descriptive. And the most common examples would be when you are answering surveys. You have the BRFSS surveys, right? The behavior risk factor surveillance surveys, or census. Every 10 years, we have a census. That's a cross-sectional study, because you're collecting all the information you can at the same time. That's a cross-sectional study. So surveys are usually cross-sectional. In this design, you have a defined population. You have to describe what you mean when you say a population. So you have a very clear defined population. Then you're collecting both information on exposure and disease at the same time. And then at the analysis stage, you're putting them into different boxes. How many of them smoked and said they had cough? How many of them said they smoked but did not have any cough? How many of them said they don't smoke but still had cough? And how many people said they don't smoke and they don't have cough? Those are your 4 cells. Those are your 4 boxes. Exposure and outcome. So epidemiology is again about 2x2 tables. When we say 2x2 tables, you have 2 rows and 2 columns. We don't count this outside columns and these outside rows. The 2x2 is this A, B, C, D. These are the rows and columns. Okay? And when you think about any study that you're doing, you have to be able to put your data in this format. You have to tell me what is your exposure. So I can say, okay, here's the number of people who were exposed. Here are the number of people who were not exposed. Then you have to tell me what is your outcome. Then I'll say, here are the number of people who had the outcome. Here are the number of people who did not have the outcome. And your 4 boxes are A, B, C, D. This is what gives you all your analytical associations. Whether it is a risk ratio, an odds ratio, a prevalence ratio, whatever you want to calculate, it's all a function of those 4 cells. If you cannot clarify in your head what those 4 cells are, you're wasting your time trying to do your PhD. You're going to sit another 3 years trying to figure it out. Doesn't matter how much data you have. If you're doing something related to population health, you have to be able to figure this out. Okay? So in a cross-sectional study, I told you we collect exposure and outcome information at the same time, right? So all we get is this last cell. That's it. We are not getting these 4 cells. We're just getting this cell. Your cross-sectional study only gives you that marginal. Here's an example of a cross-sectional study where they looked at the severity of illness and risk of mental morbidities among patients recovering from COVID. So here, do you want to take a guess as to what your exposures and outcomes are? What's your outcomes here? That's right. It's the severity of the illness and mental health. So now you start thinking, what's your outcome? What's your exposure? Okay? So the advantages of doing a cross-sectional study is it's quick, it's convenient, it's not very expensive, but all it will help you to do is to think about the potential hypotheses you can generate. It will give you some idea. You have started to see some patterns. So then because you're seeing certain patterns, you might say, maybe this is what could be causing. Maybe it is winter time and we are burning a lot of leaves outside. So that smoke might be the reason why we are seeing a lot of cough, because they're saying a lot of people have cough. So you're starting to generate hypothesis. Is burning of the garbage leading to cough? Right? So you start generating hypotheses. And think about your census surveys and your population surveys. You can look at multiple exposures and multiple outcomes at the same time. You can ask somebody, do you smoke? Do you drink? Do you go and do something else? Do you go parasailing? Or do you go and swim in a river full of parasites? I don't know. You can ask all of the exposures at the same time. And you can ask, do you have cancer? Do you have diarrhea? Do you have difficulty to see? You can ask all the outcomes at the same time. So with a cross-sectional study, you can think about multiple exposures and multiple outcomes at the same time. However, there are limitations to what you can interpret from a cross-sectional study. Only people who are alive can answer your questions, which means most of your cross-sectional studies are biased towards survivors, because people who had pancreatic cancer are not sitting here waiting to answer your question, right? So you're not going to get their information. So there is an inherent survivor bias when we talk about cross-sectional studies. You cannot talk about an exposure causing an outcome. Why do I say that? Why can we not talk about an exposure causing an outcome? Exactly. Thank you, sir. You are capturing both at the same time. You don't know which came first. You don't know the direction. So you cannot talk about cause and effect in a cross-sectional study. And any disease that's a very short period, you won't be able to capture it. So any diseases with short duration are not conducive to a cross-sectional study. Okay? And they are not the best study design when you have rare conditions. You don't want to be wasting your time trying to go and find somebody with a rare genetic polymorphism in a cross-sectional study of 1,000 people. Because you might get one, maybe. So you're wasting your time. What it can do is it can give you the prevalence of a particular exposure or an infection. And you may be able to say, well, the prevalence of smoking in Mysore Taluk was 5% as compared to prevalence of smoking in Nanjangud Taluk was 8%. That's it. That's how far you can go with cross-sectional studies. One form of cross-sectional studies is also called migrant studies. As you can see, our geopolitical environment, you're seeing a lot of war. You're seeing a lot of movement. Even without war, just in our own country, because of climate reasons, people from rural communities are migrating to the urban communities in search of jobs, right? So they are migrating out of their social environments. People are migrating from one country to another for different reasons, maybe economic reasons, maybe political reasons, maybe war, maybe climate change. So you're seeing more and more of movement of groups of people from one geographical region to another. So migrant studies, they're also called migration studies, are a form of cross-sectional studies. Okay? They basically give you clues about an association between a genetic or environmental exposure to a particular disease. So here's an example. Study of prevalence. Prevalence is not a rate, it is a proportion. Okay? For those of you, numerator and denominator, if your numerator is part of your denominator, it's a proportion. Prevalence is a proportion. It's usually a percentage. Prevalence or percentage of coronary heart disease among men of Japanese ancestry living in Japan or in Honolulu, Hawaii, or in San Francisco Bay Area in the United States have showed the highest rates among those who have migrated to the United States. So they compared men of Japanese ancestry living in Japan, Hawaii, and in California. And they wanted to see, did they all have the same risk of getting coronary heart disease? If there was a genetic basis, it should all be the same, right? Because your genes don't change. So that's what they were trying to do. And what they found, although they were all Japanese ancestry, men who were in San Francisco in the Bay Area had the highest prevalence for coronary heart disease. That means it was not the genetic basis, it was the environment that changed their risk for a disease. So these are a good example of a migrant study. So in cross-sectional surveys, people are studied at one point in time. There is no follow-up. we call those a snapshot of the population. You can measure attitudes, beliefs, behaviors, personal or family history, genetic factors, or existing past health conditions, or any other attributes. You can combine a cross-sectional survey with a follow-up, and then you can create a cohort. You did a survey and you took permission from them, say, can I call you back maybe 5 weeks from now? And they'll say yes. Then 5 weeks from now, you call them up again and you get some more information. Now you have 2 time points on those people. Now that becomes a cohort. Because now you have followed up these people in time. Okay, so you can start as a cross-sectional survey, but with a follow-up or with a function of time, it can convert into a cohort. You can also do repeat cross-sectional studies. I do one cross-sectional study today, standing at Devrajas Road. Everybody who's coming to Devrajas Road, I pick about 100 of them and I give them a survey. I go back 1 month from now, I go stand in the same place. I take the first 100 people who are willing to answer my survey on Devrajas Road. I do another survey. So I can do serial cross-sectional surveys and I can compare and see who are the people coming at these different time points. Are they the same people? Are they different people? What's the patterns? What kind of people? We can start figuring out. Those are called serial cross-sectional studies. And source of most of what we know about populations is through cross-sectional studies. Like I said, surveys are a great example. When we talk about national surveys, you're getting a representative sample of the population, and you are using complex sampling designs. I'm not going to go into that right now because of lack of time, but you have to use certain sampling strategies to sample your population for these national surveys. And then you can do it on telephone or door to door or by sending a survey in mail. There are different ways you can do it. But national surveys are logistically very challenging. Again, because we want to know the denominator. It's not how many people answered your survey. It's how many people answered your survey out of how many people you had reached out to. Without that denominator, your numerator means nothing. So don't come and tell me I got 100 surveys back. My question is, how many did you send? That's important. So make sure you have your numerator and your denominator if you're doing cross-sectional surveys. So census is a good example. Okay, so far, any questions? Because that's the most common study design you're going to use. Okay, so the next one, like I said, you do a cross-sectional and you follow them. Now it becomes a cohort study. In a cohort study, again, exposure, outcome. In a cohort study, you're starting with the exposures. How many of you smoked? How many of you did not smoke? You follow them in time. How many of you got the disease? How many of you did not get the disease? So you start with the exposure, follow them in time to see how many in each group got the disease. That is a cohort design, okay? When you think about it, what you're saying is a certain number of people in the exposed group are going to get the disease, and a certain number of people in the unexposed group are going to get the disease, right? That's essentially what our hypothesis is we're saying smoking causes cancer. So we know a certain number of people in the smoking group are going to get cancer, and a certain number of people, even though they don't smoke, will still get cancer, right? So those people who become deceased in the exposed group, that's a new case, right? That's the first time you found that they had a disease. That's called an incident case. So you have incidence of an outcome in the exposed group, incidence of an outcome in the unexposed group. You have two groups, always. When you're doing analytical study designs, you have two groups, right? Now, the ratio of the two, which is what you have here, incidence in the exposed, incidence in the unexposed, the ratio of the two is what gives you your relative risk. Okay? You keep reading these words in the journal, odds ratio, risk ratio, hazard ratio, and you don't know what that means. It's essentially the ratio of incidence of an outcome in an exposed to incidence of an outcome in the unexposed. And that's what you get with a cohort study, the relative risk. Here's an example. The association of COVID-19 infection in pregnancy with preterm birth. So what is the exposure here? COVID-19. What's the outcome? Preterm birth. Exposure COVID, outcome preterm birth. Am I making all that noise? Okay. Advantages of a cohort study. Best study design when you have a rare outcome. Exposure to a certain drug. Is it me? I'm going to hold it. Yeah. When you have a certain chemical that you're exposed to, or a certain environmental hazard, you're exposed to something. Anything rare, like tsunami was rare, war was rare, it's relative now, right? Those are rare exposures. So cohort designs are perfect for rare exposures. You can measure how many people had a disease, new disease, because you're following them in time. So you know how many of them were disease-free when they started. And you followed them in time and you found out how many of them developed a new disease or a new condition. So you can actually get information on incidence. Incidence means new cases. Prevalence means existing cases plus new cases. But only cohort study can give you incident cases because you're following in time and finding out to make sure you only get the new cases. So you can measure incidence and relative risk in a cohort design. Now think about it. When we say a rare exposure, again, example, Agent Orange during the Middle Eastern wars. They were exposed to one particular chemical, but they could have so many diseases, right? It could be a skin disease. It could be a lung disease. It could be a cancer of the gut. So you can measure multiple outcomes in a cohort study. You can look for multiple outcomes in a cohort study. One rare exposure, multiple outcomes. That's the advantage of a cohort study. And because you very clearly started with the exposure and followed people in time to find out about the outcome, you can say which came first. Did the exposure come first or did the outcome come first? So now you have a temporal basis for the exposure and the outcome. Okay, those are the advantages for a cohort study. While we can all get excited about cohort studies, I love them too, they are expensive. They are really expensive because you have to follow people in time and you have to make sure you're not losing people. Which means today I might be very enthusiastic and participate in your study. 1 month from now, when you call me, I'll say, time elapa, don't come to me. I don't have time to answer your questions. So I'm lost to follow up. Now you have a problem because your denominator is affected. You started with 100 people, you already lost me. Now you only have 99, and that's a problem. So your cohort design is very sensitive to loss to follow-up. So you have to be super friendly with your participants. Make sure you send them a birthday card, wish them on Ugadi, make sure you become friends with your study participants. I'm serious, you ask PHRI and they'll tell you. We have 99% follow-up on some studies. And that's because we do all this. We'll send calendars to our participants' houses. We will wish them happy Ugadi. When somebody comes, we make sure they have a cup of tea and we talk to them before we give you a survey. So cohort studies are resource intensive. They take time, they take money, and they are expensive. You need large sample sizes to answer questions. And again, they're not suitable for rare diseases because you have to follow them for a very long time. Think about if your outcome is cancer, do you know how many decades you have to follow a person? Those researchers are a unique group of people because they're just as old as your participants or maybe older because you have to follow them for so long, right? So you don't want to be using a cohort design for rare diseases. And here you can measure association by comparing incidence of the disease in the exposed, incidence in the unexposed, all the different exposure groups. Any questions so far about cohort designs? You see how easy this is? And you all were wondering, epidemiology, my goodness. It's simple. You just have to think in a certain way. Another version of cohort design is case control design. Case control design is not any different than cohort design. It's just the direction of your exposure and outcome changes. Now here, you're going to start with the disease and the non-deceased, what you call cases and controls. And then you're going back in time to find out what exposures they had, which means you can only do a case control design if you have fantastic data collected from the very beginning. You cannot say, I'm going to go to JSS tomorrow and do a case control study. Why? Because I don't know how good your electronic medical records are. I don't know if you have all the data. If you have good data that has been collected over a period of time, yes, case control study is a design you can consider. But if you do not have exposure information collected accurately, case control designs will not help you answer your question. Essentially, case control studies, like I said, are a version of a cohort study. It's just another way of sampling. How you sample your study participants and measure the exposure and outcome association is basically what a case control design is. And here, you remember I told you, you have to define your population. In case control design, that is the most important thing, is how you define your population, which we call study base. How you define your study base? How do you define who is this group of people that I am going to examine to see what exposures they have? Because I have these people who have the disease and I have these people who don't have the disease. So again, it comes down to your denominator, your study base. What is your study base? You have to be able to define it. And usually, all of your case control studies, they are done in they're a subgroup of a large study base, in which case many times it could be a cohort. You have a cohort of people you're following, then you can sample from that cohort. Now your study base is your cohort. Your chance of having that case come out of that population is the same as your chance of getting a control. So the cases and controls have the same chance of coming from that denominator. So that's what it is. your case control studies are done within some cohort, defined or non-defined, which what we call a study base. And that's why they say in reality, cohort and case control designs are very artificial, because you're almost examining the same thing, an exposure and an outcome, the relationship between the two. Ideally, your cases and controls must represent a fair sample of your underlying cohort, which I'm saying is your study base. So in a case control study, if you are writing a case control study, you can tell me everything about your exposure and your outcome, but you do not tell me what your study base is. That's a red flag for me. Means nothing. You're wasting your time sending that paper out. So the most difficult part of a case control study are two things, is how you define your study base. That's your denominator or your population. And how you select your controls, because this is a case control study. Cases are easy, right? It's a disease, so you know it. But how do you select your comparison group is the most challenging part of a case control study design. Like I said, your controls should come from the same study base as your cases. That means your base population from which your cases came, your controls have to come from that same base. You can't say, I'm going to take cases from my hospital at JSS. I will go out to Bamboo Bazaar and I will pick up all my controls. Unless you want to define your study base differently for me, there are two different study bases. If this rule cannot be followed, that your controls are coming from the same study base as your cases, you better have a very clear explanation why and be able to justify it. You have to have a solid rationale for it. And within a strata of factors that you're going to use to stratify in your analysis, your controls should be selected independent of your exposure. That is one of the major assumptions. So when you are thinking about your controls and how you're going to select your controls, you have to make sure that you're not inadvertently somehow connecting your exposure status with your controls. And it happens a lot of the times in case control studies. And that's what leads to misclassification. And it gives you new levels of confounding that you did not have before. So you can already see case control studies are a little bit more complex than your cross-sectional and your cohort studies. And I haven't even touched the tip of the iceberg in terms of case control studies, because this is an overview. So case control studies, be very careful. So when you think about how you represent your case control studies here, you're starting with the deceased and the non-deceased, right? Cases and controls, cancer, no cancer. And then you're going back in time to look at how many of the deceased group had the exposure and how many of the non-deceased had the exposure. So now you're saying, what's the odds of exposure in the deceased? What's the odds of exposure in the non-deceased? And the ratio of the two is what gives you the odds ratio. So your case control studies can only give you one measure of association, and that is odds ratio. Odds of exposure in the deceased, odds of exposure in the non-deceased. So what you're getting is the exposure odds ratio, not outcomes odds ratio. Outcomes odds ratio you'll get in other study designs. Because here you're starting with outcome and finding out what exposures they have, you are getting exposure odds ratios. Here's an example, risk factors for non-melanoma skin cancers in Sao Paulo, Brazil. So your cases here are non-melanoma skin cancer. And they want to see what kind of exposures they had. So they concluded fair skin, family history, occupational and non-occupational sun exposures were associated. Those were all the different exposures that were associated with having the cancer. Advantages, you can study rare diseases. Because you're starting with cancer, you're not waiting for somebody to get cancer, right? So any rare disease, you use a case control design. You can actually go and look for multiple exposures, right? Because you can look at fair skin, occupational, non-occupational, all the exposures, right? So you can look for multiple exposures at the same time. It's relatively cheaper than cohort studies, and it can cope with long latency periods because you're starting with the disease. You don't have to worry about it. Okay? Limitations, you cannot calculate risk. You can only calculate odds. You cannot talk about temporal relationships because you're starting with a disease already. You don't know whether the disease came first or the exposure came first unless you have excellent record keeping. It's not suitable for rare exposures. And there is an inherent bias in this design. And you have the selection bias because you're already selecting. You're choosing who is in your study, right? Because you're starting with deceased and non-deceased. So you have selection bias. And you're going and telling, 10 years ago, did you drink 5 cups of coffee? And you're expecting him to tell you the truth, right? That's what we do in case control studies. We go and say, 5 years ago, did you go cycling to this place? So you have issues with recall. Because if I had cancer, I'd say, I have cancer. Yeah, I did. I used to go to that place. It had so much smoke, right? And we recall things differently based on how we are feeling at that point. If I'm deceased, I might think, yeah, I did all that. And if I didn't have the disease, I'm not going to make the effort to even think whether I had 5 cups of coffee 10 years ago. So depending on my mood, I might say yes or no. If I fought with my husband, I'll say, I don't want to talk. Then you have missing data, right? So all of those biases are associated with your case control design. And your information on exposures is only as good as how it was collected. You can measure the association by comparing prevalence of exposures. Remember, it was exposures that we were looking at in the different deceased groups. Any questions so far? Cross-sectional cohort case control, case series, case reports. So we're done with all observational studies, okay? Ecological study design is a kind of observational study, but now you are not looking at it at an individual level. Now you're taking a 60,000 feet aerial view of populations. Now you're looking at a correlation between exposures and outcomes in different populations. So you have no information at an individual level. You only have information at a group level. It's usually, again, used for generating hypotheses. So it's usually one of those first studies that you do. You're looking at patterns at a very high level. It's not a very strong design. Example is alcohol sale in Mysore and NAFLD or cirrhosis of liver in Mysore. You're not getting individual levels, right? JSS hospital tells you there are about 5,000 cases and you know what's the alcohol sale rates in Mysore. It's all at population level. Smoking rates and tobacco taxes. What's the prevalence of smoking and what's the tobacco tax at a population level? These are all ecological studies. Again, when you look at the 2x2 table for an ecological study design, you are only getting the marginals, the outside row and column. You're not getting the individual cells. You're only getting group level information. It's usually at an aggregate level, at a group level. Your unit of analysis is not an individual, but it's a cluster or a group. It's used for generating hypotheses, and it is prone to something called ecological fallacy. You cannot adjust for confounding because you don't have a way to compare. But this is one of those quick and easy way of trying to start thinking, what are the kind of questions I want to ask? So it kind of gives you some clues. Here's an example of a correlation of TB and HIV seroprevalence in African countries. So each of those dots is a country. Your x-axis is HIV seroprevalence, and your y-axis is the incidence of TB per 100,000 person years. Okay? You don't have individual level. Each of those points is a group. It's a country. And you're looking at the relationship between TB and HIV. So why do ecological studies? Well, they're low cost, convenient. You cannot measure anything at an individual level because your interest is not at an individual level. You are trying to understand something at a much more higher, at a population level. It's very simple, straightforward in terms of analysis. And it's, again, like I said, it's useful for generating hypotheses. You're looking at aggregate information. Example, study of relationship between gun ownership and community rates of suicides and homicides. Again, it's all at groups. You're interested at a population level exposure, not individual, but at a population level. Same thing, you're interested in the outcome at a population level. Living in a neighborhood with high rates of violent crime can affect people's mental health. You're not going and asking each person what kind of issues you have mental health. You're looking at how many people are seeking mental health services or how many people are seeking counseling services. And you're looking at the police records to see what's the violence, what's the crime rate. It's all at group level, okay? Here's an example of breast cancer mortality and dietary fat intake. So each of the dots is a country again. Your x-axis is fat intake and your y-axis is breast cancer mortality. Here is another ecological study, chocolate consumption, cognitive function, and Nobel laureates. So your x-axis is how much chocolate you eat. Your y-axis is how many Nobel laureates came from each of these places, and each of those flags is the countries. Is there an association? Switzerland makes me think so, right? So eat more chocolate, right? That's ecological fallacy. I said just because Switzerland has high Nobels and high chocolate, does it mean each individual is smart? No, but I said eat more chocolate, assuming that yes, we are smart. That's ecological fallacy. So when you're taking a conclusion from a group level and assigning it to an individual level, that is called ecological fallacy. So the limitations for ecological studies is it seldom leads you to causal relationships. Data on other factors may not be available. There is no individual link between your exposure and your outcome. I'm going to skip this. So that is all about observational studies. I'll very quickly go through experimental because you guys are not going to be using as many experimental study designs because they are super, super expensive. They cost lakhs and crores of rupees to do a clinical trial. and most of us will not be involved for a very long time in it. The simplest of your experimental studies is a simple 2-arm parallel randomized controlled trial. What we are saying here is you have a certain population. You're going to choose a small group of that population to be part of your study. Then you're going to assign an exposure. Either they get the treatment or they don't get the treatment. Then you're going to follow them in time to see how many of them got cured and how many did not get cured. So this is the simplest of the study designs when it comes to experimental RCTs. Here's an example of a 2-arm parallel double-blind randomized controlled trial to look at the efficacy of omega-3 fatty acids for treatment of women with endometriosis-associated pain. A mouthful, but very simple. Your exposure here is omega-3 fatty acid. So a group of them were assigned to take omega-3s. A group of them were assigned not to take. They were followed to see how many of them were pain-free. 2 arms, randomized, and they were looking at outcome, which is endometriosis-associated pain. Did that omega-3 work or not work to clear the pain? You have another design called crossover RCTs. Here, you are the case and you are the comparison or the control. You are both. How does this work? You start with a population, you take a subsample of them, you randomize them to a treatment or no treatment, right? And then what you're going to do is you're going to follow them in time for a small period of time to see whether they had an outcome or not. So you'll measure the outcome. Then you'll wait for some time to wash off the effect of that exposure or the medication you've given them, right? You have a washout period. And then you're going to switch them. Those who had the treatment now will become the control, and those who did not have the treatment will be given the treatment, and then you follow them again and you measure outcomes. So you're crossing over. So you are your own control. That's the advantage with a crossover design. But it has its limitations. Not all drugs or all interventions can be tested with this particular study design because of this important period called the washout period. If your drug doesn't wash out fast enough, that effect can continue to influence your outcomes. So here is a randomized crossover trial of dietary sodium restriction for stage 3-4 chronic kidney disease. Okay, so they actually had a group of people. They had 58 adults with stage 3-4 chronic kidney disease, and then they had them exposed, and then they waited, and then they switched them. And then they looked at the outcomes. The last one that I'm going to talk to you about is a factorial RCT. A factorial design is used when you want to test multiple interventions at the same time. You want to test 2 interventions or 3 interventions. Usually in a single 2-arm, you can only test 1 intervention at a time. But with a factorial design, you can test for 2 interventions simultaneously. So you have 4 groups. You will have, again, you have the population and the sample. First, you're randomizing somebody to both the drugs, and 1 group to no drugs, and then 1 group to having 1 drug, and another group to having another drug. So now you can compare and see which of the 2, did they work synergistically? Did they work very well? Or individually, did they work very well? So you can answer those questions simultaneously. That's when you use a factorial design. So here's an example of a factorial design to evaluate the effect of micronutrient supplementation and aerobic exercise on maternal endothelium-dependent vasodilation and oxidative stress of the newborn. So there are two interventions here, where the micronutrient supplementation and aerobic exercise. So one group of women got both. One group got either the micronutrient, the other one got the aerobic exercise, and one group got neither. And then they went and looked for vasodilation and oxidative stress. So that's a very classical factorial design. So when we talk about RCTs, here you are assigning the exposure. The randomization assignment is a random allocation of exposure or treatment. The result, or it should result, in two equivalent groups of almost the same characteristics. So when you're reading a clinical trial or an RCT, the most important piece of that paper you want to read is table 1, where they describe your population of the study participants. And you want to make sure that both groups look very similar in terms of their age, their gender, all of it. That means you have actually randomized your known and unknown confounders well. So that is why a randomized controlled trial can actually give you the causal relationship between an exposure and an outcome, because of that magical thing you do called randomization. So it is the gold standard for causal inference. Advantages: least subject to biases because you're randomizing. Disadvantage: it's very expensive to do. You have to have a certain statistical analysis that you have to do. If you are using an RCT, you have something called intent to treat analysis, ITT. But many people fail to do intent to treat analysis, and that's when the problems start. You have the issue of loss to follow-up. You can have randomization issues. If randomization is not done well, your clinical trial is going to have issues, and you can see that in table 1. All exposures cannot be randomized. So there are ethical issues associated with doing clinical trials. So you have to be mindful about that. So I've talked about all of these study designs, and I've given you like an aerial view of it. But I'm assuming you have learned a lot more than what you started with an hour ago. Okay? You know now when somebody says cohort, case control, cross-sectional, case report, case series, factorial design, single arm, double arm, all of it. So you pretty much have a good idea of all of these study designs. So when you talk about a study design and you say, oh, I'm going to do a clinical trial, I'm going to ask you, you better be sure why you want to do a clinical trial. So it goes back to what I started with. What is your study question? What is your hypothesis? That will decide what study design you're going to use. And that's what drives your conclusions. So what do you want to be able to say at the end of your study is going to define what your study design is going to be. And that is going to decide or will be decided by what your study questions. So they're all connected. Okay? So I will stop there. Thank you for staying awake. I appreciate it. And I'm happy to take any questions you have.
Thank you, Dr. Purnima, for a wonderful session. So at the beginning, you told I'm going to keep it very interesting, and it was really interesting. And you touched on so many important points, which are neither given in any textbooks, and they're not given in any, typical notes or something. But you spoke with your experience, and you spoke with your heart. what exactly is required. And when we talk about study designs, people think it is to be learned only by the epidemiologists, but it is definitely everything related to study design should be known by everyone. And most of the time we conduct the studies and then students conduct the studies and then come and ask what type of study design it was. So we get to read very crazy study designs when we review the papers. Retrospective randomized control trials. Oh, something which is, which you cannot imagine. Prospective cross-sectional study based on case records. So something which can never be palatable. The moment we come to know, the next note is going to be kindly consult an epidemiologist and reconcile the study design. So on a lighter note, study design is like selecting your life partner. If you select a good one and you select a proper one, your life is going to be easy. Otherwise, it is going to end up in a mess. Exactly same with that of the research. If you select your study design correctly, your outcome is going to be, measurable. Your outcome is going to be generalizable. Otherwise, we will end up in spending so much of resources. Using a wrong design will end up in disaster. That is the reason your session on, as you aptly mentioned, you have touched the tip of the iceberg. The whole textbook of Oxford textbook of public health, whole thing talks about the epidemiology and the study designs. And, it takes a lot of time to understand and, internalize it. But what is expected to know for you, I think Madam has brought them on a very, very relevant note. Give her a big round of applause. And I request, if there are any questions, kindly you can pose some questions to Madam so that she will be able to answer from her experience. Thank you.
They understood everything so well. There's no questions at all.
So can we make one of them give a talk on study designs now?
Absolutely. Now that they know it, they should share with their peers.
Ma'am, one question.
Yes.
What if we have more than 2 groups? You were mentioning that we take an exposure, we take an outcome. One with the exposure and the disease, one with the exposure, no disease. Then you have no exposure but disease, no exposure, no disease. Like that, if we have more than 2 groups, then how do we go about it?
Thank you for that question. So when you're talking about exposure, when you do the analysis, you can actually have your exposure categorized into multiple strata, right? And that's how you use the models to look at, as compared to your referent group, if your age group is 10 to 15 years, what was the risk of the disease versus what was the risk of the disease in the 30 to 40-year-olds as compared to the 15 to 20-year-olds. So you can actually have multiple categories of exposure and you can still look at the outcome, your odds or the risks, depending on what your study design is.
Hello, ma'am. You said using the word prospective and retrospective study as a separate entity is wrong. But we have seen in many researches, and I myself have published many papers using those things, and even in good journals as well. But I still didn't understand how is it wrong using only...
Just because somebody says it doesn't make it good. Just because NEJM publishes a paper doesn't make that paper great. Okay. NEJM publishes the worst papers. Just because it's an NEJM doesn't mean that's great.
Okay.
So don't get carried away by the journal. Don't get carried away just because somebody used it. That's not a reason. You use it if you have a rationale for it.
No, but basic understanding is when we are conducting a study, taking samples which are already there in the hospital, that we call as retrospective study. And what we are actually looking in the real time and collecting the samples and processing, that we call it as a prospective study. That is what basically we understand. And
that is your definition.
Yeah, exactly. That is what I want the clarification. It's not just mine. Many of them have the same dots and they're all sitting silently.
It doesn't matter. That's what social media does for us. Misinformation and disinformation. Just because a group of people are saying COVID doesn't exist, doesn't mean COVID doesn't exist. Just because 2 million people are saying it and it's on Instagram and it's on Facebook. Don't use that as the reason for saying something is just because a lot of people are doing it, so it's okay. It's not okay. It's not okay because retrospective could be case control studies. Your retrospective could be your retrospective cohort studies. Again, the question is, what is your exposure and what is your outcome? Which one are you starting with? Are you going back in time and starting with an exposure and then coming forward and checking for their outcomes? Because they're all collected from your hospital records, but what are you starting with? Are you starting with exposure and then checking your outcome? Or are you starting with your outcomes and then going back and looking for exposures? Both are retrospective. So which of the two study designs should I interpret you are doing? So what should be my measure of association? Should it be a risk ratio or should it be an odds ratio?
So these things I can include under a cross-sectional study. For example, I'll give you an example. So if I collect FFP blocks from hospital, from pathology section, and if I know this belongs to head and neck cancers or some other cancers, then I perform say some immunohistochemistry. I know those belongs to a particular cancer, and I have all the clinical pathological characteristics of the patients. Then I'll be performing the IHCs and everything. I'll be looking for some markers. Then I'll be grouping them under some groups, and then I'll be doing the association studies. So will this be considered as retrospective or not?
What is your study question?
That's what I know. They are cancer patients.
What is your study question?
I want to know the association if these cancer patients have any kind of increased or decreased expression of a particular marker.
So your study question is what is your specific marker?
Say NRF2.
Is there an association between NRF, whatever your study marker is, and head and neck cancer? That is your study question.
Yeah. Also in between HPV positive and negative. So first we'll be analyzing.
Now that's another study question.
No.
Don't mix up things. Let's start simple. What's your hypothesis?
So hypothesis in HPV positive.
We're talking about NRF and head and neck cancer. So let's stick with that.
HPV associated head and neck cancers. I'm just giving an example.
Okay, so your outcome is HPV associated head and neck cancers. Your exposure is NRF.
Yeah,
right.
I mean, NRF2 will be overexpressed in HPV negative head and neck cancers, whereas it is decreased in HPV positive cancers. So in order to identify if the patients are HPV positive or not in the old samples, I mean the already stored samples, obviously we'll have to do either PCR or a P16IH or something. So first we'll be doing that. So my basic question is, if we are collecting those samples and I'm doing an analysis, is it considered retrospective or not?
I cannot answer that question till I ask you all the basic questions that I started with, right? What is your exposure? What is your outcome? You have told me.
Okay.
Now you're saying you're going back in time and you're collecting all this information because it's already there.
Yes.
So you have your cases, right? So now I have to figure out what is your controls.
So actually here we'll not be comparing between the cancerous and non-cancerous ones.
So there is no comparison.
HPV positive and HPV negative. Because head and neck cancer patients, either they can be having HPV virus infection or without the virus infection. So if we are dividing them like that, whether that will be a retrospective study, that's what is my question.
There is no such thing as retrospective. Please do not use those words, retrospective and prospective. Praveen, help me here.
Yeah. Everything is contextual. As Madam properly mentioned, retrospective and prospective studies, technically speaking, should not be used, and they should be used in the context. In what context you are actually describing your study design. Let me take his
yes, sir,
point.
Please.
So the exposure and outcome are your NRF2 and your cancer. Okay. So you have previously collected blocks, and at one point in time, you are assessing the NRF2 levels in that, and the patients are already having cancer. Based on that, you are going to tell whether the expression of NRF2 or whatever is more in those patients with cancer or not. Simple way of telling the study design is a cross-sectional study. Because though the samples are collected at one point, at different point in time, you are analyzing them and coming to the conclusion at one point in time. Are you all getting the point? So the point at which you collect the data or you analyze a particular sample, that determines your study design. In a very simple way, we can just call it as a cross-sectional study. And if you are having two groups here, for example, many times we take a group of diabetic patients and we take a group of non-diabetic patients, and we assess HbA1c levels in diabetic patients, HbA1c in non-diabetic patients, and we want to compare both of them. The question comes is whether we call it as a case control study or not. Because according to you, the person having diabetes is a case a person not having diabetes is a control, and I'm taking HbA1c in cases and HbA1c in controls and finding out whether there is a relationship. If we use the standard terminologies, you want to ask whether it is a case control study. No, it is not case control study. So that you need to go back in time. Do you have, that's what the reason she was mentioning. If you have a strong baseline data of your records, then only the case control study has to be mentioned. That becomes just a analytical cross-sectional study. That ends the show. So in your case also, as you are assessing NRF levels and cancer, so your study is going to be cross-section.
Yes, sir. I understood it is a cross-section study.
Yes.
Even I consider it as a cross-section study.
And you're talking about
that, So if under that, whether there is something called prospective or retrospective, that is what is my question. What exactly is retrospective? Because you have been reading about it so many places.
Time at which you start. Let us go back to the previous definitions, age-old definitions of prospective, retrospective. What people used to consider retrospective studies if your collection of data begins long time back. I'm in 2022. My data collection began in 1978. And in 2022, I will gather all the data that came from 1972 till 2022. What I did, I went back in time, started the data from back, came till this time. So you are walking retrospectively. That is the reason it was called as retrospective study. Okay, not all case control studies are essentially going to do this. Though you are doing case control study today, your data collection would have started, your data would have started some time back. But my point of argument is, you cannot call case record analysis as a retrospective study. We do not accept it. If you are doing a case record analysis, tell it is a case record analysis. That's all. Case record analysis, you told you have collected some records from somewhere and you call it as a retrospective study. We do not accept it because in case records, there is no starting point. What happened in, study related to consumption of, mother consuming diethyl stilbestrol and children developing vaginal cancer? There were many women in UK developed vaginal cancer and they tried to find out what is the cause for this vaginal cancer. And they went back to the hospitals where these women born. And in those hospitals, they checked what these mothers took. I'm telling you the strongness of data we need to have to tell it as a case control study. They went to their mother's records when they were in the first trimester. Many were prescribed diethyl stilbestrol. And they took the controls, their mothers had no consumption of diethyl stilbestrol. They came, they traced the data in forward and then they told the consumption of diethyl stilbestrol in the first trimester is associated with vaginal cancer in women, okay, their daughters. Okay, so I think you got your point. Don't call case record analysis as a retrospective study. The point is starting point. As madam mentioned, better not to use these terminologies. Epidemiology is something which is evolving over a period of, by the time she finished her talk, many concepts in epidemiology would have been refined. Okay, so what madam is trying to tell is the refined form of epidemiology which we use today. People were telling you cannot conduct a cross-sectional study in the hospital. That's what we learned, me and madam, when we were studying. We all would have learned you cannot conduct a cross-sectional study in the hospital. But hospitals are the major sources of cross-sectional studies now. Okay, so concepts are evolving. Better not to use those terminology. I think I made my point clear. Thank you.
Thank you. Thank you for saying it.
Sorry.
No. I'm glad you clarified, because maybe I was not as eloquent as you. I think one of the things what sir was saying is you need to have that high quality data that has been collected for a long time. And that's why the European medical record system is so amazing, that they have such fine data. You can actually do case control studies, and you may not have that many issues with misclassification. Because of their strong record keeping, you can go back decades into their records. I don't think we have that level of quality here to be able to say we can do that kind of studies.
So case control studies, be very careful. Always think which one are you starting with, exposure or an outcome. That will help you in deciding whether it's a cohort study or a cross-sectional study or a case control study. That's how you want to start thinking about these things. Absolutely. I'll be happy to, there are so many free online downloadable books. And the other thing is there are online courses on Coursera that you all, if you are excited and you want to learn more, you can actually go and take an online course on Coursera on study designs. There's a lot out there. Yes.
Ma'am, if a study is based on randomized control trials, will it be wrong to say that it is a meta-analysis?
So randomized control trials and meta-analysis are two very different things. Randomized control trials, your unit of analysis is an individual, right? Meta-analysis is a synthesis of studies. So your unit of analysis in a meta-analysis is a study.
So does it involve a lot of groups, like exposure?
You can have two studies and you can combine them statistically and it would be called a meta-analysis.
Okay.
Two studies. If you can get all the variables that you need to combine them statistically, and anytime you combine them statistically, it's called a meta-analysis. But your unit is a study, not individual. And your RCTs are usually at an individual level.
Thank you, ma'am.
Thank you, Dr. Purnima Madhivanan, for the wonderful talk. Now I thank Dr. Praveen Kulkarni and Dr. Kiranmayi Muralidhar for chairing the session. Now I request Dr. M.V.S.T. Subbarao sir to hand over the memento for Dr. Praveen Kulkarni and for Dr. Kiranmayi Muralidhar as a token of appreciation. We will now have a tea break for 10 minutes and let's gather again for the last session of the day.
Take pressure until the tea arrives. The tea will arrive in 10 minutes. Let's go out of the conference room and have fresh air.
Sure if I'm being introduced or not, so I can definitely introduce myself if I'm not. I'm Carl Krupp. I'm from the University of Arizona. I'm going to kind of take you on a little bit different journey here. I want to talk about two things that never ever get talked about when you're in graduate school. I'm talking today about scientific writing, and I'll be talking tomorrow about grant writing. Now, it's really amazing because most of us survive in academics based on the papers we write, the papers we publish, and the grants we get. But there's this crazy process, and it pretty much goes on around the world, and that is that we learn all the science, but they never teach us what we need to do to actually survive as academics. And this, I'm not sure how true it is here in India, but I can tell you quite honestly, you don't get promoted, you don't get tenure, you don't get salary increases unless you publish papers and unless you get grants. So today, even though it's late in the afternoon, I want you to turn your chairs toward me and listen, because this may be one of your only opportunities to actually talk about something other than the science that you're involved in on a day-to-day basis. I am, I'm an assistant professor, and I've been doing research for 16 years, but I have focused very heavily on refining the way that I write papers and submit papers and think about papers, and also refining the way that I submit grants. And that's made me much more successful in my field. And I wish early on I had somebody who could have sort of mentored me into the process of doing this, but it's not something that happens anywhere. And it certainly doesn't happen in the US. So let's bring up the slides. How do I get the slides back? I'm gonna turn that off. Okay. So I'm going to do something here. Okay, for some reason, I'm not on the. Okay, I'm going to do something today. One of the things that, one of the challenges I had when they said we'd like you to come and just talk about sort of best practices for manuscripts and grants is that I teach a whole course, a semester course on grant writing and one on scientific writing. So there's an awful lot of material there. And one of the things that we don't get a chance to do now is to actually work on things together. So one of the things I do with my students is we actually practice all of the concepts. So I had to kind of think about how I was going to do this, because in general, these kind of skills aren't taught with didactic lectures. So you see this star right here. What I'm going to do is I'm going to stop on the slides that have a star like this. And I'm not going to go on until somebody answers my question. So you seem to be warmed up here. You were really, really, really reticent not to answer questions this morning, but it looks like you're a little better off. But I have to tell you, we're going to be here all night unless somebody answers the question when one of these stars come up, okay? Okay, go ahead. Yeah. Yeah, what's going on? Okay, so I'm going to take you through a couple of processes here, and I'm not actually going to talk. There's something going on with this. Is there do you have a slide changer?
Yes.
Yeah. Okay. What we're going to talk about, okay, is not necessarily how you're going to write a paper. I'm going to take you through everything you have to do before you write a paper, okay? And unfortunately, what most people don't teach you is that you have to do an awful lot of work, okay, before you get on your word processor and you start writing paragraphs. And the thing is, most people go and they just start a grant and they try to write it all the way through. Okay, it's the wrong way to write. It's the wrong way to think about it. It's actually the wrong way to pick your topic. Okay, and so what I want to talk about today is how you take, you want to go to the next one? How you take your paper, okay, from the very earliest part, okay, where you've got an idea, or you've got a professor with an idea and he's told you you better have an idea. I mean, it could be either way. But the point is, you want to write a paper, okay? And the question is, what's your idea? How did you pick your idea? Okay, where did it come from? Okay, did it come from your data? Are there other better ideas that are in your data? Okay, then we're going to talk about the audience. We're going to talk about the journals you select, and we're going to talk about what an editor wants, okay? And an editor is the guy who's going to stop you in your tracks, okay, unless you give him what he wants to get, okay, when you send in a manuscript. Okay, so you can work your way through all of these obstacles if you think about them beforehand. Next slide. Okay. Now, the problem most of us have, and I'm a perfect example of this. I do a big study, and I have a study right now called the truck driver study. Okay, I did it here, and we looked at heart disease in truck drivers in Mysore. Okay, and we recruited them in union halls. Okay, and we have about 1,000 truckers, and we really put them through a pretty exhaustive evaluation of their cardiometabolic health. Okay, now what happened there? No, don't go to the next one. Yeah, go back. So here's, okay, can you back it up? Okay, yeah, so here's the problem, okay? I've got this mass of data, okay? Now the thing is, there are 100 different ideas in that mass of data, okay? Some are easier to publish, some are harder to publish, some are more interesting, some are less interesting, okay? All of them may have a home, but when you're a student, okay? You're a PhD and you're trying to get your first paper in, okay? And it's gonna make a difference in whether you become a postdoc or not, okay? You have to pick the right idea out of all of the ideas that are there in your data, okay? So we kind of want to talk about how do you do that? What do you want to pick that's going to give you the best chance to get into a good journal and get your manuscript published? So go on. Go ahead. Oh. So the way that I think about ideas, okay, when I'm sitting there with my data and I'm looking at it and I'm thinking, okay, I got to get a paper out, okay, I want it published in the next 6 months. There's an important reason for that. And let's say I'm coming up for promotion, okay, and I have to have this paper. Well, a good research topic has, no. This is going to throw me off. It really is. How do we, is there some other way to do this? Okay. A good research topic has 3 qualities: specificity, novelty, and relevance, okay? And when you're looking at all those ideas you have in all that data you're looking at, and it could be that you're in a lab and you're in your mentor's lab and you're doing a study and you've been collecting data on something. I can guarantee you that there's more than one paper in the data that you collect. Okay, I can guarantee it. You may not think so, but once you sit down and you think about it, you'll find those ideas that will be successful manuscripts. Okay, you go on to the next one. Okay, so what is specificity? Specificity refers to using words that are concrete and concise rather than general, abstract, or vague. Okay, so most of the ideas we have that we want to turn into papers, they're very vague ideas, okay? So my suggestion is sit down with a paper and a pencil and write the idea down, okay? Because that's when the rubber hits the road and you actually figure out, is this a good idea? So go on to the next one. It's like working in slow motion here. Okay, so here's the star up in the corner. Now, we have 3 ideas here, and we've just learned that specificity, okay, says that it should be concise and detailed. Okay, so who can tell me here which idea is the best? Should be pretty, okay, go ahead, tell me why. Why is it the best?
Because it has the name, from which year to which year the data has been collected, from whom it has been collected, what is the disease, and also what is the amount of increase that is there. It includes all the concise information.
Okay, so it's concise and it's comprehensive. It has, you know, very specific details, okay? Now, the second one is interesting and might make a paper. It may be, the paper may be there, but it may not be ready yet, okay? The third one, I'd kind of stay away from that one, okay? Because it's just too vague and it's gonna be a struggle to take that to a paper. Go ahead, next one.
He's saying that he will connect this to the Wi-Fi portal if you give a minute's time so that it will get.
Okay. Okay. So while he's doing that, so we can speed this whole thing up, because I don't want to keep you here all night. How many people have published here? How many people have publications right now? So 2, 3, 4, 5, 6. Okay, that's pretty good. Okay. Was it hard to publish? What was hard about it? Reviewers? You can't blame reviewers. Okay. Reviewers just have a job and they have a really specific list of things that they look through. And I've been a reviewer before and it's a thankless job. Yeah, it's completely thankless.
The rejection rates are much higher than the acceptance. Can we say papers?
Well, rejection rates are high, and there's a good reason for it because there's a lot of really bad manuscripts out there. That's why they're so high. Because the thing is, a journal editor wants to publish your paper. Okay, believe it or not, there's a profit model here. Okay, when he gets your paper, if it's a spectacular paper, okay, that his readers are interested in, he wants to publish your paper. Okay, so he's not looking to reject your paper. I mean, you get this when you first start in writing papers, you get this idea that everybody is against you, right? You can't do anything right. But the truth is that they really want to publish your paper. And if they're rejecting it, they got a good reason to do it. And you know, so you got to think about it. Well, let's see. Okay, are we back? Okay, cool. So can you back up one and we'll just figure out where we are exactly. Okay, one more. No, you got to go back. I think you're going forward. Back, two. Is there a way to do that? Go back. I want to make sure I don't miss anything here. Okay, I think we're good. So if you want to just go to the next one, we'll be fine. So we've got specificity. Oh, next one. Man, this is a tricky projector here. I don't know what's going on here, but okay, the next one is novelty. Okay. Novelty is the quality of being new, original, or unusual. And this is a problem here in India. Okay. You know, a lot of times what happens is you get, you're assigned a project that isn't new or novel or original. It's been done before. And if you look in the literature, you'll find it's been done before. So here's what you have to do when that's the case. You have to find a new angle. You have to find a new way to describe something that isn't novel so that you can make it novel. Okay? All those ideas are in the research that you have, okay? But they take extra work, okay? You're going to have a huge problem if you take a paper. If you look in PubMed and there's 10 papers on there and they're pretty much the same thing, when you send that paper out to an editor, he's going to look at it and say, God, we already published 2 of these, right? So why should they publish a 3rd one? Okay, so don't bother to do that, okay? You have to have novelty before you actually write the paper, okay? So go on to the next one. Okay, so this is a really interesting case here, and it's from the same exact data, okay? And one idea is novel and the other isn't. Can somebody give me a guess about which one is novel and which one isn't? Come on, somebody loud so I can hear. Yes. Okay, so this data had data for, or this particular study had data for 3 doses. Okay, to be honest with you, everybody's published on 3 doses. And there's now new interest in whether one dose might be good enough. So here you have the data right there, and you could publish a paper that would be novel, okay? And it's the same data, right? So I know there are publishers out there right now, editors out there right now that want to know what the outcome is for one dose of quadrivalent human papillomavirus vaccine, okay? And the fact that you're doing it 6 years following it is going to make it really exciting for you, okay? Now, relevance. This one's kind of tricky because it has to do with you understanding who you're addressing, okay? Relevance is about knowing your audience and being able to predict if they will care about your idea, okay? Now, relevance is a relative thing. So what happens is, in one journal, your article may not be relevant because that's not what they do. That's not what they're interested in. That's not what they publish. In another journal, the same exact paper will be relevant. Okay? So you have to look at your audience, and you have to decide, when I'm sending in a paper, okay, is the journal I'm sending it to actually going to feel that it's relevant to their readers? Okay, go ahead. Okay, just push it. This way. Oh, there it goes. So I have to do it over my shoulder here, it looks like. Okay. Okay. Come on. Okay, so here's a star here. So now there's lots of interest in seniors right now because we're all aging, and there's a lot of journals, journals out there run by publishers and editors that are aging too, and so there's part of the concept of relevance, okay, is about how you present, okay, the idea as well, okay? So it's not just either it's relevant or it's not relevant, but have I pitched the idea, okay, have I pitched the paper in a way that's relevant, okay? And one of the ways we do that, okay, is by thinking about how the idea, okay, can be made more exciting, okay, more relevant for the editor that we're sending it to, okay? Now, when you look at these, if you were an editor that was kind of interested in exercise, okay, maybe a little bit interested in older people, okay, which one would be more relevant? Why? Well, I can tell you it's a lot more exciting than most older people don't like exercise, because that's pretty obvious. I mean, most people don't like most people in general don't like exercise, and old people aren't any different than everybody else. But the last one gives us an exciting idea that we can intervene, okay, with something, and we can increase, okay, their activity levels and their quality of life. So we've taken an idea in the beginning there, and we've added enough that it's now relevant to an editor. So go ahead. Oh, here I got it. Now I can't blame them. Oop. So, geez. I know there's a way to do this that will make sense eventually. Okay, so one of the other things you have to think about when you're thinking about your idea, okay, is where does the topic fit, okay? And this is important because the way that journals are organized is in categories, okay? And these categories are pretty general, okay? But you have to figure out for yourself when you're thinking about your idea, you know, what pot does it belong in, okay? Okay, when I go out and I look for a journal, I know I'm going to have to look through these categories. Where does my paper fit, okay? Where am I aiming? So the other thing to think about when you're writing a paper is there's a different slant on every topic, okay? Your paper, okay, might be most relevant to a surgeon. It might be most relevant to a researcher. It might be most relevant to a policymaker, okay? It might be most relevant to a pharmaceutical company, okay? You have to decide, okay, who your audience is before you write the paper, okay? Most people make the mistake of writing the paper and then trying to figure out who to pitch it to, okay? These are all things you do before you set your pencil to paper and you do anything. So when you look at a topic, okay, there's lots of ways to think about where, oh, God, this is crazy. Man. Where it belongs, okay? What pot it belongs in, okay? And maybe we're just taking a title here, okay? And we're trying to say, okay, where does this belong, okay? Where do I send it? Who's the audience? Who's the journal? Okay, who is best going to be excited about this idea? Now, prognostic significance suggests that it's a clinician probably, right? Could be a clinician, okay? Then we get down to skeletal muscle index and systemic inflammatory index. Could be a researcher, right? It could be a clinician still, but I haven't seen clinicians use that level of measurement typically in a clinic, okay? So it could be a clinician researcher in a tertiary hospital, okay? With lots of equipment, okay, and lots of ideas. It could be somebody who's interested in cancer, or more specifically breast cancer, right? It could be a surgeon or, you know, a surgery journal, right? So how do we decide? Anybody? Seems like a lot of options there, right? So what's the best one? Probably not. It's like when you go to a restaurant and they say on the outside, Chinese, Indian, Thai. And you think to yourself, oh, that's not going to be good. Right? Because they're trying to do everything. They're probably not doing anything well. I wouldn't go for multidisciplinary. Any other ideas? I mean, how do we've got a couple of options here. The cancer? Why?
Dealing with the or the breast cancer, that may be more specific.
Yes. So in general, okay, when you're working with this kind of problem, okay, you got to see what excites you. Okay, that's my own sense of it. There's no magical thing about this, okay? If you're excited by breast cancer and you see that there might be an audience out there for a manuscript on breast cancer, then go for breast cancer. Why? Because it excites you and you're going to do a better job writing a paper, right? Okay, the only point I'm trying to make here is that you have to select your audience before you do this. Because if you select your audience before you write the paper, the flavor of the paper, the language that you use, the organization that you use in the paper will reflect the choices you've already made. Okay, there you go. So here's some titles. Let's start with the first one. Anybody? Where does that belong? Could be your paper. Where does that belong? So some kind of hospital journal or something?
Infectious disease.
Infectious disease, possibly. Yep, because it's hospital infections. Yes. Anybody else? Epidemiology? Maybe. Yeah, but there's also prevention, control, and surveillance too. I mean, my guess when I look at this topic is that it probably, it has the flavor of policymaker in it, okay? I mean, that's how I would look at it. And it's the audience that's going to be most interested in it is going to be big picture, dealing with hospital issues, okay, at a macro level, right? So if I was going to pitch that, I'd probably find a journal that would be focused on hospital policies, whatever that is, okay? Let's try the next one. Anybody? These are always the worst because we have this terrible, terrible situation we're in kind of as researchers. The world is organized in our bailiwick into organs and diseases, right? I mean, everybody asks you, what's your disease, you know? Or what's your organ? Are you working on heart disease? Are you working on kidney disease? Okay. And we tend to organize things that way. But when you get when you're looking at metabolic pathways in cancer cell survival, what you have to do is you have to pick the most important one. Okay, when you focus it. That's usually how I do it. And cancer cell survival says to me, I need a cancer journal. Okay, even though there's some metabolic, data in this particular, data set, and it might be interesting and it might appeal to people that are into metabolic pathways. That's not to say you couldn't do it that way, but that's the way I would do it probably. What about the next one? An imaging journal. That's probably radiology. Yeah, it could also go into a cancer journal as well. That's possible. But I think the fact that you're starting out there with imaging strategies it probably says to me that somebody like a radiologist is going to be most interested in that. Okay, what about the last one? Probably policy level. Yeah. Yeah. Okay. So we don't have to overcomplicate this. The only thing that I want you to think about is as you look at your title, think about it. Think who would be interested in this? Who would say to themselves, this is an article that belongs in our journal. Okay. You have to ask yourself that before you send it anywhere, even before you write it. So and this is, a few people have published here, but there's lots of talk, okay, about what journals you should pick and why you should pick them and why you should target them and which ones are better and which ones are worse. And there's lots of ways that you can look at them, okay? And I'm going to make a simple argument for new authors, and that's that you forget about all this stuff, okay? I know it's kind of sacrilegious, but I'm suggesting that there's a simple way that you can take care of all of this stuff and not worry about it as a new author. Boy, what do I do with this? Oh, it just, okay. I did it again, okay. How do we get to the next one? No, I'm missing something there. Okay, so I seem to be missing a slide here. So here's the trick, and we'll talk about it a little bit later. How the journal is indexed, okay, is really important to you as new authors, okay? If it's in PubMed, for instance, or Medline, they take care of all of this stuff for you, okay? Because they won't index bad journals, okay? And as a new, up and coming author, okay, you don't want to have to chase down a journal and see if it's a predatory journal. I mean, you can do that, it's possible. There are resources out there. What you want to do is you never want to publish you want to pick the index that's important for your particular field, and you never publish anything outside of that index. For me, it's PubMed, okay? And I early on, I made a mistake, okay? And it was I had come to India, and this was probably 15, 16 years ago. And I wrote a paper and I submitted it to a journal that wasn't indexed in PubMed. Well, there's another thing that happens to articles that aren't in the index in your profession. They disappear forever, okay? Everybody in my profession looks at PubMed, okay? And when they ask me how many papers I've published, they really mean how many papers do you have indexed in PubMed or Medline, okay? So the best thing you can do for yourself, okay, to protect yourself so you're not sending your paper to some, less than worthwhile journal is just don't send anything outside of the index that's important for your profession. So I have some just general things that you ought to think about when you're thinking about a journal. A big deal now is whether a journal is subscription or open access, okay? And we'll talk more about that. There's value to both models, but they're very different, okay? Then you want to ask yourself, is the journal general or is it a specialty journal? Who are the journal's readers? Where is the journal indexed? How often is the journal published? How fast is the publication process? And I put this in parentheses because I hear it all the time that people want us to publish in high impact factor journals. That is not a choice you're going to have as a new writer, as someone who's only got a couple of papers out. No matter what you do, you're going to end up publishing in a specialty journal, okay, with a medium to low impact factor. Why? Well, it's because you don't have the standing, okay, to go into JAMA right now, okay? And so you shouldn't worry about it, okay? What you're going to do if you go only after high impact factor journals is you're going to go after more general journals that are less concerned about your specific topic because those are the ones that are high impact, okay? And you're going to get rejected a lot and you're going to get discouraged okay? And there's nothing worse than discouragement, because what happens with submitting articles is that it sometimes takes 3, 4, 5 times, okay? And then revising it. So avoid discouragement where you can. Pick a journal. You don't have to go after BMJ right now, or JAMA, or Nature, or Cell, okay? Because there's a number there, okay? That's a self-defeating behavior for a new, relatively young, scientist. So. Okay, so there are 2 different models really for journals now, and the ones that are going to be the easiest to publish in are open access journals. And the reason that open access journals are going to be easier to publish in is because you're paying the bill, okay? Now that doesn't mean they publish bad papers, okay? There are very reputable journals that are open access, and they're just as hard to get into, and they will reject just as many manuscripts, okay? But the thing about it is that you're going to have to pay the bill to publish in there, okay? Now, I rarely do open access. I do it infrequently, and I do it only when I have a grant budget to do it, okay? Otherwise, I always go to subscription-based journals, okay? Because if they publish, I don't have to pay the bill. I mean, it's when you're early on in your career, you don't want to pay the bill if you don't have to, right? That's the whole idea. So I pretty much avoid them. I will always look through, and there will be tons of open access journals. Now, there are some exceptions to this, and we'll talk about them a little bit later. In general, okay, open access journals are going to charge you between about $1,300 and $2,500. Big money, okay, really, really expensive. Now I had one open access journal that published a paper I wrote when I was living in India here, and they actually published it for free because it was from their list of developing countries. Okay, now this was 15 years ago, and I'll bet you it's not as developing now. I'll bet you there's fewer discounts for that. But it is possible that some journals will give you a huge discount because you're in India. Okay, and you should check that out. Okay, the other thing is students often get waivers and discounts as well. It's still expensive though. If they give you 50% off $2,500, it's still $1,200. That's a lot of money, but it's better than $2,500, So. This whole issue of whether it's a specialty or a journal is really important, okay, for you. In general, okay, the specialty journals are going to have lower impact factors because impact factors are based on the number of readers that a journal has. And if you're in a really narrow area, okay, then there's going to be fewer readers. It's just like that. Now, the advantage for you, though, in publishing there is that everybody you know that's in the same business and does the same work that you do is going to read that journal, right? So you're publishing in a place where your colleagues see the work that you do. And that's important. Okay. So your field is there, okay? And they're reading those papers. So there's real value in that. And I would not be concerned about the impact factor. I would rather, as a young scientist, be thinking about how do I make my name in my field? Because that's going to take you to the next level. So if you publish in New England Journal of Medicine, that you're a really tiny fish in a really, really big ocean. you know? However, if you publish in, I mean, a good example is, community nursing. I'll bet you start to recognize the people who are writing the articles if you're in community nursing, right? Because it's a small world, So when you think about the problem of, do I want to be in a general or a specialty journal? In general, it's going to be harder to be in a general journal because you're not going to have very big appeal if you have a very narrow focus, right? And then secondarily, if the journal is in your field, and I have one journal that I published multiple times in, just because I know everybody. I actually know the editors. I go to conferences with the people. I know all these people. So it's a natural place for me to publish because that's my community. So think about it that way as well. Okay, when you think about who are the readers of different journals, what I noticed and this really something that you should avoid. Journals like New England Journal of Medicine are actually published by a medical society in Massachusetts. And medical societies and associations are mostly doctors, okay? And they're mostly clinicians. And clinicians have a very, very narrow view of what they're interested in. Now, if you're a clinician, I don't mean that in a pejorative way. I'm just saying that if you're doing molecular work in a laboratory on a very obscure, biochemical pathway, chances are the average clinician is not going to be interested, right? So if you look here, what you know is you have to ask yourself, are doctors in a medical society going to be interested in my topic, right? Okay, in this case, this is kind of interesting. This is a professor of chemistry who started the annual review of biochemistry. Okay, this is clearly an academic journal, right? So as you look at these things and you say, are those my people? Because that's where you're sending your paper to, your people. Okay? If you're not doing that, then you're just wasting time. This is exciting. When I push this button, I never know what's going to happen. Oh, there you go. Oh, here. Oh, this was out of place. So this is the issue of indexing. Okay, I was telling you. So I stick to PubMed. Okay, and I check PubMed. Okay, before I send it off, I find a journal. Okay, and I look in PubMed. And if it's not in PubMed, then I don't submit there. Okay. Now there's lots of different indexes, and depending on the field you're in, the papers in your field may be indexed in one or another. Okay, so I'm not saying everybody should use PubMed. What I'm saying is you should use the appropriate index for your papers. But what you want to do is make sure that everything that you publish is in a journal that's indexed, okay, in that index for your particular field. There it goes. So there's a couple of simple ways. I told you I use PubMed all the time. A lot of times if I'm being lazy, okay, let's say we take the Indian Journal of Sexually Transmitted Diseases and AIDS, okay? And I've got something on sexually transmitted diseases and it's in India. And so I look at the journal, seems to be a good fit. Now I just want to know, is it indexed? Okay, the way I want it to. Two ways I can do it. One is I just put the journal in there. What that will bring up is any articles that were published in that journal, okay? And if nothing comes up, that means it's not indexed there, okay? The other way is there's something called the NLM catalog, and I can put the journal in there and it will actually give me a description of the journal and where it's published, okay? And I can tell where and when it's indexed, okay? So this is a big one for students, okay? When you're in your fourth year and you need a paper, it has to be out, you're gonna be applying for a postdoc. The frequency of publication is really important. The best frequency would be, like 52 weekly issues, because that means, you can submit it and it will be, there's enough issues in the upcoming year that if they accept it, it will be published fairly quickly. Okay? So that's why you check the journal frequency. Now, it just happens, they say the frequency is 10.95 issues a year. For practical purposes, usually it ranges from one issue a year to 52 issues a year. And sometimes you can also find them, they're published on a quarterly basis, on a biannual basis, okay, or an annual basis. Those are the ones that are most important. Now, most pretty legitimate journals will actually tell you about how long it takes to publish, from the time that you submit to the time that it actually goes online. So that that's kind of how I look at it. When, in general, okay, once it's online, it'll be indexed and I get credit for it. So sometimes what happens is it'll go up online and then they'll wait 2 issues until they have some space that corresponds to the length of the article. And then it will actually go into the journal. But for me, that doesn't matter because it's already up on the web, it's on my CD, and it's indexed. So those are the issues that are important to me. But if you look at these different journals here, JAMA says the recording in progress. For an initial editorial decision is 3 days. So what you'll find in general, and JAMA is a good example, they really turn if they don't like your paper, you know about it in like 2 days. So at least you're not waiting, okay? I mean, that in a way, if you decide you want to try for a really high impact journal, they're usually pretty good. I mean, you send it to Nature, you'll probably get it back in 24 hours, the rejection. That's true with JAMA, that's true with regular journals. So at least you're not waiting around thinking, are they going to publish my paper? You'll know pretty quickly. And then once they accept, I think it's, in general, it looks like 31 days is the average for submission and acceptance, and then 15 days from acceptance to online publication. So +1, 24 weeks from submission to publication. That's a long time. And if you're desperate for something because you've got to show it on your CV, I don't think you should send it. Probably not a good call. Okay. BMJ, 8 to 10 weeks from submission. It kind of, in general, okay, when I think about getting a paper in and getting it published, I would say the process probably takes about a whole year. In general, I mean, I've had papers that have been published in a month. but I've only had like one in all the years that I've been doing this. You know, in general, I would expect when you're thinking about what do I need to have for this publication, give yourself a year from the time that you actually write it and submit it for publication to the time it goes up and it's indexed. Okay, and again, I just explained what an impact factor is. Basically, the more people who read a journal, the higher the impact factor. And they have a citation formula. Okay, that means in other words, when people read it, the way that science works, you know, if you're doing a paper, if you're doing a grant, okay, well, manuscripts mostly. You look at the literature, okay, you find an article, and then you cite it, right? Okay. Well, you can't cite it if you don't read it. Okay. So what happens is journals that are very highly read, like big medical journals, okay, are cited more often because people see the literature and they cite it more often. That's just the way it works. So in practical terms, the way it works is general journals always have higher IX because they reach a larger audience. The newer the journal, the lower the IX. Usually it takes a while. It takes a couple of years for a journal to kind of slowly move up. Okay. And new authors are more frequently published in stuff. So it just makes more sense. Now I want to talk about the editor. And the editor is just a guy that, just like you, he went to college, he studied English or communications. He went to work for a big journal, and he sits in a little office and he has a computer and he sits there all day and reads manuscripts. Okay, you got to feel sorry for this guy. I mean, that's tough, you know. But you know, I was trying to tell you before, you want to have a positive attitude about it, the journal editor, because not only do they do very hard work, but they actually care about their journal, and they're usually pretty good at what they do, and it's a difficult job. Okay, so they're the ones that get all of the manuscripts, and they select those they consider suitable, and then they send them for peer review. Okay, and this is the process that you have to get over. The biggest hump, okay, is that hump right there. That's called editorial triage. And what it means, okay, is that the editor, when he's gotten your manuscript, okay, has asked himself a couple of questions, okay? Is this manuscript in the scope of what our journal does, okay? Is it a topic that fits into our readership? Is this something our readers will be interested in? Does the article have a clear message? Is it original? Is it important? Is it true? Is it relevant to our readers? So the most important one is that one. The rest of them are somewhere way down the list. And the second most important is that one. So you can get through triage, okay? And nobody tells you to do this when you're publishing. I tell you, when you're a student and you're publishing things, nobody says to you, "What are you going to do to get through triage?" Okay? You just keep noticing that the manuscripts, you send them out and they come back. You send them out and they come back, right? Okay. Well, there's a way to actually deal with this, so they don't come back. Okay, and the easiest way is to do a pre-submission inquiry, to write a convincing cover letter. And that's something that most people completely neglect. It's an afterthought. It's like, oh, dear Mr. Editor, I have an interesting manuscript for you. Hope you like it. Sincerely. Okay. That will get you anywhere. And they're much more important than you think they are. Editors actually read them. Okay. You have to have a compelling title. And that's something that I've really struggled with. I don't, no more joke titles. They don't work, okay? Wordplay doesn't work, okay? There's a whole bunch of things that don't work, and I think I've tried them all. But, you know, the title is very important, okay? And then the last thing is the abstract is super important. Because if an editor is actually going through a lot of manuscripts, sometimes that's the only way he'll make a decision on your paper is on the basis of the abstract. I don't know if we're going to get through. Do we have time here? Are we okay? So pre-submission inquiries are pretty simple. It's just an email. But you have to have several things in it that are important. A personalized greeting, if you're looking at a journal and you could find the editor's name, send it. It's much better to say, "Dear Mr. Smith," okay, than to send to who it may concern. Okay. So the more personalized it is, the better off, you know, you're going to be. The better off your results are going to be. You have to really tell them why you think your article is a good fit for their journal. Okay. That takes some thinking. Okay. Look at their journal, look at their readers, look at the other publications that are in their journal, and convince yourself that you're a good fit. If you can convince yourself, you can convince an editor. Explain why your findings are important to the journal's reader. I mean, if your findings aren't important to the journal's readers, then it's not going to be published in there. Part of this is a relevance thing. Okay, if the readers are the wrong readers, you know, obviously it's not going to make any sense to them. But we've already talked about this. You really have to think about who you're sending it to and why they should want to read it. And editors are really slaves to their readers. Okay. Now it could be that you get a mean editor, but in general, that's not the way it works. They will publish anything they think their readers will read. Explain why your findings are important. Thank the editor. Just be nice. Okay. And no flourishes. Say something like sincerely at the end. Nothing fancy. Okay, and then there's a cover letter. And again, what I was telling you, what happens in a busy journal editor's office is that they're working on lots of things. Okay, they will, and you know it yourself. If you're really busy, what do? You scan everything, right? Even your emails, you just scan the first couple of lines, and then you say, I'm going to go on to the next one, and you scan that one. Okay, that's the way they are. Okay, so your cover letter should highlight the key findings of your study. It has to convince the editor that this study is of interest to his reading or her reading. And there's a bunch of information that they're going to ask for, and there's statements like, "We have not sent this to another journal," you know, or this has never been published in a journal before. There's specific things that you know, go look at the requirements, okay, in the journal. They'll tell you we need this language. So you got to make sure in the cover letter that all of that language is in there. If you can get it in there, you want the editor's name in there, you want the journal name in there, because they like to hear their own name. Okay. You want your manuscript title. You want to tell them what type of an article this is. Okay. You want to put in the submission date. Again, what you're doing is you want to give, if that guy's in a hurry, you want to give him everything that he needs as easily as he can possibly get it. Okay. And then again, there are things that each specific journal will ask for, and you just make sure that you know what they are and you put them in there. Now, when we come to titles, there's a lot of debate about this. And there's a lot of research on titles. Should you use a colon? Should you not use a colon? You know, in general, okay, they should be concise. They should tell you something about the article itself. They should contain words that are important in the fields and are related to the topic. Okay, they should interest the reader. Okay, so you got to have a hook in there. You know, it's got to be somewhat interesting. You got to get your audience to actually, you know, look down and start reading the article. They shouldn't be overly sensationalized. I think a lot of times younger authors, they'll put things in there that will raise the eyes of the editor. You make claims that probably they don't, you know, deliver in their paper. So you just got to be careful to not be too sensationalized. And then there are really specific instructions about, you know, title length and subtitles, and some journals have just recently had, you know, what they want is they want you to show the methods in the title. You're going to have to know that for the specific journal. You know, they may want to have that it's a systematic review and meta-analysis. They may insist that go into the title, or a randomized controlled trial. It has to go into the title. You're going to have to know that. You have to go to the journal and have to make sure that there aren't requirements. There's a few different ways you can look at a title. Okay? It's worthwhile to look at this. Titles can communicate, or titles, there's 4 different ways that they can work. They can communicate the general topic. They can point to a specific subtopic of a general topic. And I'll show you each examples of these. They can state your study's findings. You've seen this before, right? There's a debate about whether you should tell the reader too much, but there's an awful lot of successful manuscripts out there that say what actually their study shows. In this case, habitual sleep durations and subjective sleep quality predict the quantity of white matter in the human brain. And then titles that state the methodology. So sometimes, especially in engineering journals, actually mentioning the technology is important to your readership. So let's just talk about this. So remember what they are. There's a title that's about the general subject. There's a subtopic of the general subject. There's study findings. There's methodology. Let's take the first one. Anybody? What is that? It's a mess. So, what kind of title would work for that? Just think about it. You're pitching your paper. Your title is kind of your hook for the editor. If the editor is at the Journal of Molecular Epidemiology and Evolutionary Genetics of Infectious Diseases, what kind of I've given you the 4 different types. What? And you could do study findings. What was the other one? Subtopic. Yeah, it's probably subtopic. What about the next one? Journal of Oncology. Probably general cell, right? What about the last one? Okay. So the only thing here is that that you have to realize is there's actually a science to figuring out your type. And one of the ways that you can think about this is go look at other titles too. Go on whatever index you're used to looking at, PubMed or whatever, look up papers. And then if they've been published and they've been published in good journals, emulate the title. But think about your title. It's not an afterthought. It's a forethought. It's something you do before you send it to a journal. Okay, so finally, I'm just going to talk about this because structured abstracts are used practically everywhere. Okay? And they're really simple. But you have to do what you have to do in a structured abstract. They have 4 parts: the background, the methods, the results, and the conclusions. So the background is really structured. I mean, these things, once you start writing them, become so mechanical that you'll do them in your sleep. But you have to, the first sentence has to be a succinct statement of the issue. I'm giving you an example. The widespread acceptance of COVID-19 vaccines is crucial for ending the current COVID-19 vaccine or pandemic, right? So it's general, it's big issue, okay, and it's always this way. Okay? You start with the general and you get down, and as you move down, you get more specific. You have to have a brief summary of the research problem, given the problem here. Understanding the reasons for hesitancy to vaccinate is critical to designing effective interventions to increase vaccine acceptance. Okay? The third element is a summary of the current research. And this can be one sentence. Okay? Studies suggest that public hesitancy is primarily driven by poor healthcare access, low literacy, disinformation, and barriers to access vaccination. A description of the research gap, okay, and then a description of the purpose of the study. Now, this is what should be in every single abstract that you write. You should never not have any of those elements. It's really easy. You look at the list and you say, do I have that element in there? And if you don't, put it in there. I mean, it's not a mix and match proposition. Okay? Abstracts are always written the same way. Okay? When editors see that you haven't put one of these things, they're going to send your manuscript back. So it's, I mean, what I would do is, for me, I just have a checklist. And I look through the checklist. And if I've got all those elements in there, I'm fine. And just be mechanical about it. Make sure you put in what you need to put in. And that's the background. The methods. Okay. All you're doing is describing what you did. And the best way to do it is to use your W's. When, what, who, and why. And they should be in there. If it's a methods section, then you should tell when, you should tell what, you should tell who, you should tell why. Again, very mechanical. And finally, results. You're just describing what happened. And this is your field. So when you're writing, you know what results are important. Now, the only thing I would suggest when you're putting results in an abstract is don't give everything away. Pick a few things that you think are important and don't give everything. You still want them to read your article. They don't have to read, if they just read your abstract and they got the whole point, then why would they read your manuscript, right? So be selective, put a couple of juicy facts in there that you found, and then leave the rest for the manuscript. Finally, you have a conclusion section. You have to tell the editor what it means. What does this mean? What is this about? What is the point of all this? That's what you're trying to tell them. What this author said is our overall findings revealed considerable pathway changes in lung metabolism and asthmatic lungs, including energy, amino acids, and lipid metabolism. Nice, concise sentence that says, I'm not going to give you the facts, I'm going to tell you what it all means. Okay. One of the things that you should always consider when you're sending your manuscript into a journal is the editor isn't necessarily a specialist in your field. You really have to tell him what's important about this paper. That's your responsibility. That's not his responsibility. First. I'm almost done here. Yeah, well, I don't know. Let's see. So the abstract qualities, an abstract lives or dies on whether it's complete, whether it's concise and short, right to the point, okay, whether it's understandable. So don't use a lot of jargon in your abstracts. Because again, your reader may be somebody that's not in your field. So make sure that they understand what you're talking about. Is that it? Yes. Man, I got through it. So any questions? Come on, you're all going to have to publish. Go ahead.
So, there are many journals are there, which means it will be a good idea to see the journal. Like, I have an idea, or I'm doing a project in my PhD, and while I am completed all my results and everything, the research I have completed, and the articles I am writing. And should I put all the articles, means all the results, as you said, that we should not put all the results and all the things, means which the art journal needs, that type of data only we should give them. So if I want to put everything of my project work inside the article, because you told sir that a good means audience. Yes,
no, what I said is don't put it all in the abstract. Okay, you put it all in the article. Don't worry about that. What you don't want to do, so the only thing that's indexed, if I go to PubMed, I just see an abstract. Okay, what I want, I mean, what you should do is it should be interesting and have just enough interesting data in it that I'll say, I got to read that article. And then I go and read the article. You don't want to give away everything in the abstract. That's all.
So actually my question is like, first, or whatever ideas I have got to write my project, or for the idea to get, and like if I'm writing a review article, so for the review article, I have to go through all the articles. I have to be a good audience. Then only I can be a good writer, or I can impress the editor. if I'm a good audience. So on that basis, I'm telling sir, if I read all the articles and if I get an idea, and with those published articles, if I think of writing another article which is not similar to them but with some of the novelty, and then if I try to publish, but sir, every time when I'm doing this, like all of my review articles is getting.
You're going to get old doing it the way you're doing it.
Yes, sir.
So
if any
my suggestion is
yes, sir.
What I usually do is I look at who I'm referencing. Okay, so I'm writing the manuscript and I've got a bunch of references in there. Okay, I go and look at the references. Chances are there's a journal that will publish this in the references. Okay, there's another part to that too, is if you're sending a manuscript off to a journal, make sure that you reference some article in that journal. Okay, sometimes I've had to go back and just find some reference and put it in there. Okay, but they look to make sure that you're referencing that journal. So the 2 points are, see who you're referencing, okay, and look at those journals and see if they're appropriate for what you want to submit. Okay, don't spend too much time investigating the journal. You'll get so many manuscripts back, and then you'll have to start all over again, right? Just do it the easy way, okay? And also, like I said, if you're sending it into a journal, make sure that you go find some article that you can reference from that journal in your publication, because editors care about that. So, anything else?
I just wanted to ask, if we have lots of data, we would still, some of the students have lots of data, and we want to decide how to publish it. Is it to publish 2 different papers or one all together?
Well, what you're looking for is an interesting idea. Okay, if there's 2 or I could guarantee you in the data you have, there's 2 or 3 good ideas, good strong ideas that will make manuscripts. Okay, and some may be easier to publish than others. Okay, so there's this big debate, okay, about whether you're a sausage publisher or you're a lumper or a sausage maker. That's what they say. So what you do is you put every bit of data in one big manuscript, or you turn it into sausages and you send out like 10 different journals. You send out parts of I don't think it's a relevant discussion. I think the whole thing is, does this idea stand up on its own? Is it worthwhile? Will a reader read it and say, I'm glad I read this article? Okay, so what I would do is I would look at the data and say, okay, let's just assume that there's 4 or 5 good ideas in here, and then list those ideas, and then pick the best ones.
But sometimes what we do is, in order to substantiate a set of data, we will set up another essays to justify the previous data. So under those circumstances, how do you?
I'm not sure I understand.
Let us say we are doing a set of Western blocks.
Yep.
We have got some good results.
Right.
And that is not enough for a good publication, just for a high impact journal. So we want to do something else, not to prove that it is that. So how do you?
Well, the first thing I would do is, I would tell you, I'm really suspicious of high impact journals. So I'm just not sure that's a good distinction. The question is, how much data, you know who else is publishing articles in your field, okay? Go look at the articles, see what they're publishing, see how much data they have in there, okay? You're in a community and you're all publishing in that community, and there are standards, okay? So if you look at 10 articles and this is how many Western blots they need, then you're going to have to give them what they need. Yeah. So it's really a community standard. You're part of a community of scientists that are all doing the same work. Go look at what your colleagues are doing. That's the best standard, I think. I know that's not a satisfying answer, but I don't know your field, so I can't tell you specifically how many, of this or that it should be in there. But I can tell you that there are standards in your field. I know it, so anybody else? Yeah, go ahead.
Then they have a set of we start working towards that. And really not sure it's really going to get through. So when is the right time for us to really, how do we get, as a student, when is exactly the time? Because like she said, maybe we have some data and we get through or not, and they might require some other additional experiments with us.
Well, let me answer you this way. With a graduate student and with a scientist, yesterday is the day you should have started, okay? And it's always yesterday, okay? Because you have to publish, okay? And you can't sit around and wait for the perfect moment. And you're never gonna have the perfect manuscript, okay? And you are always gonna get rejected. So sitting around and saying, oh, I've got to wait until I have the perfect data, the perfect amount, for the perfect journal, you're doing it wrong. You should, if you think you've got some data that's interesting in your field, try to publish it. You'll find out pretty quickly if there's not enough. And then you can go back and fill in. But don't wait around for the perfect whatever. It's never going to happen. And there's going to be a day when your professor looks at you and he says, you only have one paper. Okay. That's the day you should avoid. That means you have to start now and start publishing now and start thinking now about what you want to do. But don't assume that there's only one paper in your data either. Because everybody assumes that. And I don't know why they do, because there's lots of good ideas in the data you have. You just have to look for them. So may I know the difference between site score and impact factor? So the science score? Yeah, it may be something that I don't run into in my.
It is mainly from Scopus. Scopus uses site score, and Web of Science uses the impact factor.
So again, it's an index issue.
Yes. Roughly about the same.
Yeah, so I don't use Scopus, so I would.
They use different indices, right? So the calculations are different based on what is it, but roughly they reflect the same thing, right? Approximately the same thing.
But again, I wouldn't spend too much of your head on that. Find a good journal and put your papers in. That's what you want to do. Don't sit around and say, oh, I've got to target this New England Journal of Medicine. All you got to do is get it back in like one day. Well, go ahead, send it out. At least it'll come back quickly. You don't have to worry about that. But don't, you're wasting your energy. Figure out who publishes what in your field and then do that. Okay? Because nobody's, as a young scientist, nobody's going to be beating you up for impact scores. I guarantee you. Nobody's ever asked me. All they want to know is how many manuscripts have you published? Now I know if I went to Harvard or something, maybe they'd start asking me. So far, they haven't been asking me that. So I don't know why people put so much energy into it. Just find a good journal, send your stuff out, Anybody else?
Carl, I would like to add a few things from what you said. Thank you very much. It was a very nice presentation. What I would add on is always read more from the journal that you want to publish. So read a lot of articles from the journal that you want to do, where you intend to publish, because you know what level of articles they get accepted or what kind of research gets accepted in that particular journal. Right? And it's important that you relate your research work to that kind of level. So not relate with impact factor. I also agree with that. Relate the journal to the level of research that you do, probably a little higher, so that, even if there is a chance of getting rejected, you might still move on further. Then I agree with Carl that citations are more likely. Uh, Publish in your specialty journal because there is more likelihood that it will increase your citations, and that is more important than actually impact factor. Getting a New England Journal of Medicine rather than any GM, I would publish in a biochemistry, my specialty journal, because that is where people are going to read and cite me, and there is more readability. And don't get carried away by journal impact factors. There are many journal impact factors. Some may say universal journal impact factor, global journal impact factor, galaxy impact factor. There are many things that have come in now. And many times in the university, we find that a very good research gets published in a journal of very low impact, with a predatory journal, just because the author or the scientist got carried away by the impact factor which was put on the journal, 6.5 impact factor. And then I say, my God, this is a predatory journal. And then you can't do nothing. It's published, it's on the open domain. You can neither retract the article nor you can kind of so good research especially gets lost and does not give any weightage neither for the person who did the research, nor the faculty, nor the university. Right? So don't get carried away. And of course, time frames, Carl did mention. Being an editor myself, I know that there is always some soft corner once the time gets delayed. If it is more than 6 months, I would, as an editor, start looking at, if there is no reason to reject an article, I will again see that there is more likelihood that the article gets accepted. So the longer the duration, the sweeter, I should say. Then review a database. Most of the times, being an editor, we ask the authors to kindly provide names of reviewers. As an author, we think that we don't need to fill this and we just go out. But I tell you, as an editor, it's important because we are building a reviewer database and we want reviewers who will review our papers. The time duration for an editor is lost because of the reviews. 1 month we send it for review, we don't get a response, we send reminders, again 15 days, we don't get response, and then we have to withdraw the article from the reviewer, send it to a fresh reviewer. And this review process sometimes takes 3 years. And the author is patient. I, as an editor, have to be patient, and we have to complete the review process. So please send us reviewer database. Sometimes we might send it to the same person whom you recommend, provided they do an unbiased review, and we are convinced that it's an unbiased review. There is a chance. So don't miss out that reviewer database. So always add to the reviewers suggested reviewers, because it adds to the reviewers database. We might not send this article, might send the other articles. Then always understand what the editor wants. Important thrust area, like there was COVID-19, everybody was important. People wanted to know more about COVID-19. So look at if it's a thrust area and tell the editor that it is something which is very important. What he said was right. And often, based on my experience, I say that conclusions are very important in your study. Sometimes you draw broad conclusions, and when we look at the conclusions, we say that, okay, no, this is a very broad conclusion and it's not so the conclusions should be on what study you have done, right? Don't generalize the conclusions. This is some of the points which I just wanted to share. Thanks.
Thank you all. I have one question, Dr. Krupp. It was a great informative talk. More of a philosophical. So most of the journals now, from subscription, they go into open access. And people from developing countries like India, we're having hard time even paying for those open access. What you feel, and how about Dr. Purnima and Dr. Sir? They feel about, in US, how are they feeling about open access journals?
I'm really bugged about because these guys are publishing things online. I'm not even printing.
Yeah, that's totally business it is becoming.
When I think about what they're doing and the value they're adding, I think it's terrible that they're charging $2,500 for it. On the other hand, let me just throw this out there. We have tried. So there's this other phenomenon, which is we do a lot of research in India, and sometimes, the system conspires so that
researchers that want to actually read our papers in India can't, because they're not part of a subscription service, or they don't have a university portal that they can get into the journals. And so sometimes we've gone to open access journals because we know it will be published in a form that anybody that wants to read it here in India can access. So there's a benefit to the model, in the sense that you can make the information more widely known. But I think right now it's almost predatory. It really bothers me a lot. And it used to be, like I said, I've had luck a couple of times getting, I think, especially graduate students, it doesn't hurt you to approach a journal and say, can you waive the fee because I'm a doctoral student. It's not to say that they're going to do it, but we've had a couple of articles, I think, that have been published without any fees, because we've just made a personal appeal to the journal. It doesn't work all the time, but I'm saying it doesn't hurt to do that. And again, I think where I've kind of come down is I still try to go to a subscription journal first, and second and third, and there may be a point where maybe my department has some money and they say, oh well, we're willing to pay the fee. There's always something, and so I'll use an open access journal, but it's painful. It's painful, and I think it's exclusionary. I think what happens is a lot of younger scientists don't get the opportunities to publish sometimes that they should, because of the way the system is working right now. So, Purnima?
I'm dead against paying. Why should I pay for a business model where the editor is making me review papers for your journal, and they're also making me pay to publish my article? Why? So what I do is I don't go to open access journals. I go to subscription journals, but I make sure that my papers are accessible to all, because I only publish in PubMed index journals. And because I'm an NIH researcher, all of my research, if it is funded by the NIH, is open access. So all my research is accessible to all of you, because I only publish in PubMed index journals. So if you go on PubMed, you'll find my article. So what you want to do is think strategically. Who do you want your papers to reach? And try to aim for those journals. And there are very few open access, just like the PLOS series, the BMC, those are credible ones. But most others are predatory journals, and you don't want to be associated with them. Because once you're associated with them, that sticks. That just sticks with you, because your paper is on their website, and then everybody is going to judge you based on that. So you want to be very careful how you choose your journals.
Anybody else? Any last questions? All I'd say is just publish. Just get your papers out there and make your mistakes, and and get published. I think the worst thing you can do is to sit around and worry, does anybody want my paper or not? You'll find out quickly enough. And you got to have a thick skin. When it comes back, Purnima, I love publishing with her, because we've had papers that came back 8 times. And the thing is, it wasn't because it was a bad article. It was because we couldn't find a journal that was the right fit for that particular article. It just wasn't, it was a little different. And so we could, but Purnima just keeps putting them in, and eventually they get published. And you have to think that way. Don't hang back and think, oh, unless I can get into Nature, I'm not going to publish this article. Figure out where your colleagues are publishing. Okay, that's the other thing. Figure out who your community is. Ask your colleagues, where are you publishing? Because they're doing the same kind of work you are, right? And they're getting published. So, but don't expect that the first time you send it out, it's gonna go right into publication. I don't think it's ever happened to me. I don't know.
I had it twice.
Okay, there you go.
Vaccine journal, both times, vaccine.
Yeah, so it may happen to you, but don't depend on it. That's the way you should look at it is it's going to be tough. I'm gonna especially once you get into peer review, they may come back to you with a lot of revisions. Just be nice. Thank them profusely for their observations and tell them how you're going to change your paper. Okay. Don't get frustrated and then get short-tempered with the editor because it won't help.
Yeah, if I can add to that, when you're responding to the reviewer's comments, I've seen a lot of times you respond and say, oh, go refer to that page in the manuscript, or go and do this, or see the above comment. Don't ever do that. You want to make it as easy, literally stupid easy for the reviewers. If there is a question, you answer it. Whatever you have changed in the manuscript, copy that piece, put it right down here in quotes and say on page, this is how we have changed it. So that they don't have to go searching for anything. It has to be right there in front of them. And if you're repeating something, repeat it. Don't say, go and look at question 2 from reviewer 1. Don't do that. Because you're saying, I'm not important enough. You're telling me to go look at reviewer 1's comment. No. You just write it again, because there's no word limit for you for responding to the reviewer's comments. So make your response to the reviewer's comments as impersonal, objective, but make sure you are giving it to them in their face. Don't make them look for things.
Yeah, the other thing that, just from experience, I know, all this pain will be forgotten. As soon as it gets published. So you just got to get through to that point. Just forget it. Thank you.
Thank you.
Thank you.
Thank you, Dr. Karl Krupp, for the informative session. Now I thank Dr. Prashant Vishwanath and Dr. Rajesh Kumar T for chairing the session. I request Dr. Prasanna Santekadur to felicitate the chairpersons with memento as a token of appreciation. Yeah, we have come to an end of the first day of INDO-US workshop 2022. So I hope all of the sessions were insightful to all the participants. We'll begin our second day of workshop tomorrow early at 8 a.m. So I request all of the participants to finish their breakfast early by 7:30 and gather in the boardroom exactly at 8 a.m. so we can begin the sessions without any further delay. Thank you all.